Welcome back to IMAT Tutor. This is a channel where we take the entire IMAT syllabus one lecture at a time and build it from the ground up until nothing on that paper can surprise you. If this is the first video of yours, everything is free, everything is in order, and everything is written specifically for the IMAT and not for a general biology course. Let us begin. One thing that separates people who score well from people who don't. They practice against real traps, not just facts. I have put together a biology question bank, 873 questions covering all 25 units of the syllabus. What matters is not the number, it is what sits under every answer. You get why the correct answer is correct, and then you get the part almost nobody writes, why the option you were tempted by is so attractive, and what the examiner was testing when they put it there. Every question also points back to the exact section of the biology guide it came from. So a question you miss becomes a 5-minute fix instead of rereading a whole chapter. Pay what you want, free lifetime updates. Link in the description. Right now, as you sit and watch this, you are contracting muscle. Your diaphragm is pulling down so you can breathe. Your heart is squeezing about 70 times per minute without you asking to. The tiny muscles around your eyes are adjusting focus on the screen. Your gut is quietly pushing your last meal along. 4 completely different jobs, 4 completely different speeds, and every single one of them is being done by the same fundamental machine. A protein called myosin pulling on a protein called actin powered by ATP. That is the beautiful thing about muscular system and cellular physiology, where we would see how a nerve impulse becomes movement, and we will also discuss the molecular machinery that shortens a sarcomere. Nature did not invent a new engine for the heart and a different one for the biceps. It built one engine and then tuned it. It changed the wiring, changed the calcium supply, changed the regulation, changed the fuel, and out came 3 tissues that look and behave nothing alike. So when you learn this lecture properly, you are not memorizing 3 separate systems. You are learning one system and then learning the 3 ways it gets modified. Now let me tell you why the IMAT loves this material so much. The biology section gives you 23 questions. Human physiology is a reliable slice of that. And within physiology, muscle is one of the most heavily represented topics years after year. The reason is not sentimental. It is that a muscle question can test 4 things at once. Ask a student what happens to the H zone during contraction, and you are simultaneously testing anatomy, testing whether they understand the sliding filament model, testing whether they can visualize a structure they have never seen, and testing whether they fall for the obvious wrong answer. That is enormous examiner value from one question, so they keep writing them. And here's the part that should make you sit up. Almost every muscle question on the IMAT is built around a specific, predictable misconception. Not a random fact you might not have read. Rather, a misconception. The examiner knows exactly what a half-prepared student believes, and they write the wrong answer to match that belief. Which means the wrong answer will feel right, it will feel obvious, it will feel like the one you can pick quickly and move on. And this matters enormously because how IMAT scores you. A correct answer is plus 1.5. A wrong answer is minus 0.4. A blank is 0. Look at what that means in practice. If you confidently pick the trap answer, you do not just miss out on 1.5. You go backwards. The gap between the student who knew the trap and the student who fell for it is 1.9 on a single question. Get 4 of these wrong across the paper and you have handed away nearly 8 points to students who simply knew where the traps were buried. So that is the promise of this lecture. I'm not going to read facts at you. Every single time we hit a place where the examiner has laid a trap, I'm going to stop, name the wrong belief out loud, take it apart, and replace it with the correct version. By the end, you will not just know muscle physiology, you will know where the examiner is standing waiting for you. Now let us see the map on the screen and see the road ahead. We will open with the three muscle tissue types: skeletal, cardiac, and smooth muscle. This is the orientation slide. We establish who is voluntary, who is triaded, how many nuclei each has, and which structural features belong to one type and one type only. This slide alone kills a whole family of exam questions. Then we go inside the muscle fiber and study sarcomere ultrastructure. We will study Z-line, M-line, A-band, H-zone. This is the vocabulary that everything else depends on. If you cannot picture a sarcomere, you cannot answer a sliding filament question. And the IMAT asks sliding filament questions constantly. We will build it piece by piece. Next comes the regulatory proteins, troponin complex and tropomyosin. In our diagram, you see myosin over here and actin. Apart from that, we need to know about troponin and tropomyosin, and we will see the diagrams in the later part of our lecture. And these are the gatekeepers that decide whether contraction is allowed to happen at all. Understanding them is what turns calcium from a random ion into a switch. Then we will talk about the engine itself, the cross-bridge cycle. It will have 6 steps, it will have ATP roles and power stroke. And in the diagram, you can likely see that 6 steps are shown here. I will tell you in advance that 2 of these steps are where students consistently lose marks. We will spend real time here. We then move upstream to the neuromuscular junction. We will talk about ACH or acetylcholine, nicotinic acetylcholine receptor, acetylcholine esterase. You see here, this is where a nerve talks to a muscle. It is also where several famous poisons and one famous autoimmune disease do their damage, which is exactly why examiners like it. From there, we go to T-tubules, triads, DHPR and RyR coupling, and those are represented in this segment of our diagram. And this is the bridge between electricity and movement. It is also, in my view, the single highest value slide in this entire lecture, because the skeletal version and the cardiac version work by genuinely different mechanisms and the exam knows it. Then comes sarcoplasmic reticulum and sarca-mediated relaxation. This part represents it. Relaxation is not the absence of contraction. Relaxation is an active, energy-consuming process, and students who do not know will lose marks. We then zoom back out to the whole muscle with motor units, size principle, summation, tetanus. You see in this part. How does the nervous system decide whether to lift a feather or a suitcase using the same muscle? That is what this slide will answer. Then we will talk fiber types, represented here in this segment. Type 1, type 2A, type 2X. Why a marathon runner and a sprinter have physically different muscles and which properties travel together. Then we will look at the ATP sources: phosphocreatine, glycolysis, oxidative phosphorylation. 3 fuel systems you see here, 3 time windows, and a very common exam question that simply asks you to match a sport to a system. Alongside that, we cover muscle fatigue mechanisms, where I will have the correct interpretation of something you were almost certainly taught in school and which is now known to be wrong. Then smooth muscle, calmodulin and MLCK pathway you see over here, which is a completely different regulatory route, and the contrast with skeletal muscle is where the marks live. Then will come cardiac muscle, intercalated disc, CICR, plateau phase. Why your heart can never cramp, and why that is the reason you're alive. And we finish the physiology with resting membrane potential and the muscle action potential. This graph describes it. This slide will talk about the electrical foundation underneath everything we will have said, which also transfers directly to the nervous system lectures. And after that, finally, a walkthrough of 10 IMAT-style questions solved out loud, every wrong option eliminated by name. That final section is where all of this turns into marks. In this slide, we have actually given the whole picture in a nutshell. And I promise you, after the lecture completes, you will be able to produce everything from memory, which is required for the exam, rather than just memorize some random facts. Because IMAT will not ask you random facts, rather it will ask you the correlations between the systems. Now read this box with me, because it governs how you should use everything that follows. The IMAT marking scheme is 1.5 for correct, negative 0.4 for wrong answer, and 0 for blank. And it applies to every question in this lecture. No matter what the marking scheme becomes in the future, this will hold true always. So let me translate that into strategy. A blank will cost you nothing. A wrong answer will cost you nearly half a point. So the question is never simply, do I know this? The question is, have I eliminated the trap? If you can rule out 3 options and you are genuinely torn between 2, answering is mathematically sensible. If you have no idea at all and you are choosing among 5, the expected value of a random guess is slightly negative, and you are better off leaving it. But here's the thing I really want you to take from this lecture. Guessing strategy is what you fall back on when preparation failed. Preparation is what makes guessing unnecessary. Every trip I name in the next 3 hours is a question you will never have to guess on again. Let us start building now. Now comes muscle tissue types. To understand clearly, we will look at our canvas. In our canvas, you see 3 tubes side by side. The first tube, you see, this tube is long, straight, and thick, and it runs the entire length of your upper arm. That's why an arm image is given here. The second one is short, stubby, and it branches like a twig, connecting to its neighbors at both ends. The third is a thin spindle, fat in the middle and tapering to points at both ends, fat and tapering. And it is wrapped around a hollow pipe like a rubber band. You see, this is the hollow pipe given here. And it wraps around the pipe like a rubber band. Shapes are different. 3 shapes, 3 jobs. The long tube is biceps, built for pulling hard along one line. The branching one is hard, built so that when one cell fires, every neighbor hears about it instantly. And the spindle that we set is wrapped around your intestine, built to squeeze slowly and hold that squeeze for hours without tiring. Now here's the wrong belief I want to kill immediately, because it costs student marks every year. Many students carry an unspoken rule in their head that goes, striped equals under my control and smooth equals automatic. It is a tidy rule, and it is also wrong, and the examiner knows you have it. Your heart is striped. You see this image, this is cardiac tissue. And the heart is beautifully, unmistakably striped. Exactly like your biceps under the microscope. You see over this image, this is a skeletal muscle section. Remember, your heart, this one, you have precisely 0 voluntary control over it. Try right now to stop your heart by deciding to. You cannot. So striping and voluntary control are 2 independent properties that happen to overlap in skeletal muscle and then come apart in cardiac muscle. The moment you separate those 2 ideas in your head, a whole category of exam question becomes trivial. Why is heart striped at all if striping is not about voluntary control? Because striping is simply what you see when contractile proteins are lined up in neat repeating rows. Heart muscle needs to contract fast and hard and in a coordinated direction. So it uses the same organized architecture as skeletal muscle. On the other hand, smooth muscle does not need speed. It needs slow, sustained T-force. So its proteins are arranged in loose, diagonal, crisscrossing bundles rather than neat rows. And here's an image of the smooth muscle that we were talking about. You will see that it has no repeating order. There is no repeating light-dark pattern seen here. As there are no order, there will be no stripes. And that is the entire explanation. Quick note before we go further. The anchor slides for this lecture are free on my store. They are the same slides on screen right now, so you can follow along instead of pausing to write everything down. Link is in the description. Good luck. One more thing to build before we go to the slide. Look again at the long skeletal tube. I mean this one. That single cell can be 30 centimeters long. Now ask yourself a question nobody asks. How does one nucleus control 30 centimeters of cell? And the answer is that it doesn't. During development, dozens of precursor cells, you see here, which are called myeloblasts, line up and physically fuse with each other, merging their cytoplasm into one giant cell. See here, keeping all of their nuclei. So a skeletal muscle fiber is not one cell that grew. It is many cells that merged. And because the middle of the cell is packed solid with contractile machinery, that means the proteins, those nuclei got pushed out to the edges. You see one nucleus here, another here, and they stay right up against the membrane, not in the center. That is why skeletal muscle nuclei are peripheral. This one is peripheral too. This is not a random fact, a consequence of how the cell was built. After the description of the canvas phase, we can safely move to our anchor to match the narration with the slide. First of all, muscle tissue is contractile tissue generating force through actin and myosin interaction. Notice how the definition is written. It does not mention stripes. It does not mention control. It does not mention shape. It mentions actin and myosin because that is the one thing all three types share. Everything else is variation on the theme. And we got three types classified by striation, control, and cell morphology. Looking at the key features, we will see skeletal muscle is voluntary, triaded, multinucleate with peripheral nuclei, and has long cylindrical fibers. That is our first tube that we seen. And every part of that description we just built from scratch. Voluntary because a somatic motor neuron has to tell it to fire. Striated because the sarcomeres line up. Multinucleate with peripheral nuclei because myeloblasts fused and the machinery pushed the nuclei to the edge. Long cylindrical fibers because that is the shape you need to pull along a single axis. Cardiac muscle is involuntary, triaded, uninucleate, or binucleate with central nucleus and branched cells. Involuntary and striated together, exactly as we said. And notice, uninucleate or binucleate with the nucleus in the center, not at the edge. These cells never fused. So they kept their single nucleus sitting in the middle where a normal cell keeps it. And branched because branching is how you would build a network rather than a rope. And finally, smooth muscle is involuntary, non-striated, uninucleate with central nucleus, spindle-shaped. Cells will be there in smooth muscle. Involuntary and no stripes because there is no repeating order. One central nucleus and that tapered spindle shape that lets cell pack tightly around a tube. Let us walk the table properly, row by row, because the exam pulls single cells out of exactly this grid. Striation, yes for skeletal, yes for cardiac, no for smooth. Two yeses and a no. Voluntary control, yes for skeletal, no for cardiac, and no for smooth. One yes and two no's. Compare those two rows against each other and you can see the trap forming. The pattern of yeses is different in the two rows and cardiac muscle is where they disagree. Nuclei per cell. Skeletal has many peripheral nuclei. Cardiac has one or two central. Smooth has one central. Fusion versus no fusion. Cell shape. Long cylinder for skeletal, short branched for cardiac, spindle-shaped for smooth. rope, net, and spindle. Intercalated discs. No, yes, and no. This row is pure gold for the examiner because it is the only row where cardiac is the sole yes. We will build this disc properly in slide 12, but plant the flag now. Intercalated discs are cardiac and only cardiac. T-tubules, abundant in skeletal, fewer, wider in cardiac, and in smooth muscle, they are absent, and they have caveolae instead. We built T-tubules in slide 7. For now, note that smooth muscle simply does not have them and uses little membrane pits called caveolae instead. Calcium sensor, troponin C in skeletal, troponin C in cardiac, and calmodulin in smooth. Read that row twice. Skeletal and cardiac use the same sensor. Smooth uses a completely different one. This is one of the most reliably examined facts in the whole topic. Speed of contraction, fast, intermediate, and slow. And fatigue resistance runs almost exactly opposite. Low to moderate, high for cardiac, very high for smooth. That inverse relationship is not a coincidence. First contraction means burning ATP fast, and burning ATP fast means running out sooner. Your heart has been contracting since before you were born without a break, and it can do that precisely because it is not trying to be fast in the way a sprinter is fast. Here's the trap written again in print. Cardiac muscle is striated and involuntary. A stem describing striated involuntary is cardiac muscle, not skeletal. Skeletal is the only voluntary muscle type. Let me show you how the exam weaponizes this. The stem will not say the word cardiac anywhere. It will describe a tissue which will have properties like striated, involuntary, cells joined end to end. Then it asks where you would find this tissue and offers you the biceps, the wall of the small intestine, the wall of the heart, the wall of the bladder, and the diaphragm. A student running the rule striped equals voluntary sees striated, thinks skeletal, and picks the biceps or the diaphragm. Both are wrong. The word involuntary in that stem was doing all the work, and it was placed there deliberately. So train yourself. Whenever you see striated and involuntary in the same breath, your hand should already be moving toward cardiac. The second trap is about smooth muscle. Smooth muscle lacks troponin entirely. It uses calmodulin and myosin light chain kinase for calcium-dependent regulation. Only skeletal and cardiac use the troponin tropomyosin system. This one is a favorite one because the question can be asked from either direction. It might say, which protein is present in skeletal muscle but absent in smooth muscle? Or it might say, which molecule binds calcium in smooth muscle? Same fact, two doors. Students who learned troponin as the calcium binding protein without the qualifier walk through the wrong door both times. So say it precisely from now on. Troponin is the calcium sensor in the striated muscle. Calmodulin is the calcium sensor in smooth muscle. We will build the full smooth muscle pathway in slide 11 and you will see exactly why the difference exists. Now we come to the sarcomere anatomy. And this is the most important slide in the lecture for building mental pictures. So put your pen down for 2 minutes and just watch me demonstrate the diagrams step by step. First, I point at two vertical lines, one over here, another here, and they're some distance apart. You can call them fence posts. From the left fence post, I hang a row of thin ropes, green colored ropes you see, which are reaching rightward toward the middle. 1, 2, 3, 4. From the right fence post, I hang another row of thin ropes, which are reaching leftward toward the middle. The two sets of ropes do not touch in the center. There is a gap. Now floating in that central region, I lay down a set of thick bars. You see the blue color bars? These thick bars are not attached to either fence post. They are held in place in the middle by their own separate anchor, the gray bar you see. And this line, the gray bar, is running down the center. And crucially, these thick bars are long enough that their ends slide in between the thin ropes. You see, some portion of the thick bars are inside the thin ropes. So near the center, you have thick bars alone, and further out, you have thick bars with the thin ropes woven between them. That means this region only has thick bars, but in this region, you have both thin and thick bars, and that is the sarcomere. The fence posts are Z lines. The thin ropes are actin. The thick bars are myosin. The central anchor is the M line, and the whole thing repeats end to end thousands of times down the length of the fiber, like carriages on a train. Now the crucial question, when this thing contracts, what actually happens? And here's the wrong belief, and it is almost universal. Students imagine the filaments are shortening. By filaments, I mean this actin, myosin filaments. Students picture the ropes, I mean the actin, myosin filaments, crunching up like a concertina. You see this? When you play this instrument, they crunch up together. Or sometimes they imagine it is shrinking like an elastic band. It feels right. Contraction means getting shorter, so surely the things inside get shorter. But in reality, they do not even by a nanometer. The thick bars stay exactly the same length. The thin ropes stay exactly the same length too. What changes is how far they slide past each other. That means this region that you can imagine. Think of it like your fingers. Hold both hands in front of you, as seen in this image. Your fingers are spread, fingertips just touching. Your hands are far apart. Now interlace your fingers and push your hands together, seen here. Your hands are now much closer. Did your fingers get shorter? Of course not. They slid past each other. Your fingers are the same length they always were. And that is what contraction basically is. The myosin head, the blue bar, reach out and grab the actin, the green bar, and myosin pull it inward. As a result, the Z lines, they come closer to each other. And that is called sliding. So filaments slide. Filaments never shrink. Hold that image because everything about the bands follows from it. If the thick bars never change length, then the region defined by the thick bars never changes size. If the thin ropes or the actin are being pulled inward, then the region containing only thin ropes must get smaller. And if the thin ropes are invading the central zone where the only thick bars used to be, then that central zone must shrink and eventually vanish. 3 consequences, all from one idea. You do not memorize the band behavior, you derive it. If the later portion felt difficult to you, then stay tuned. We're going to unravel all the information in the following slides. Now the slide defines sarcomere as the contractile unit of striated muscle, spanning from Z line to the next, fence post to fence post. And note the phrase striated muscle, which means skeletal and cardiac, not smooth. And its resting length approximately is 2 to 2.5 micrometers in human skeletal muscle. 2 micrometer is roughly a quarter the width of a red blood cell. These units are absurdly small, and there are billions of them working in parallel. That is how a machine made of jelly lifts a suitcase. Now the key features. The thick filament is myosin 2, 10 to 15 nanometer diameter, anchored at the M line by myomysin, which is another protein. As we seen in our canvas, this myosin is our thick bar, held in the middle by a protein called myomysin at the M line. Note it is anchored centrally, not at the Z line. The thin filament is F-actin polymer of G-actin monomers, anchored at the Z line. 2 names for one thing that confuse students. So let us fix it. G-actin is a single globular actin bead. F-actin is what you get when you string thousands of those beads into a filament. G for globular, the single bead. F for filamentous, the chain. The thin filament is the F-actin and it is anchored at the fence post. Titin This is the largest protein in the body. Spans from M line. If you draw M in the center, it will span from M line to Z line, and it acts as a molecular spring. Titin is the unsung hero here. Picture a coiled spring running from the central anchor all the way out to the fence post, as we have drawn a rough diagram. And this titin is threaded alongside the thick bar. When you stretch a muscle, titin stretches and pulls back. Without it, the thick filaments would drift out of center and the sarcomere would be pulled apart under load. It is also genuinely the largest protein your body makes, which is a fact examiners enjoy. Now comes nebulin. Nebulin rules the length of the thin filament and runs alongside actin. If titin is the spring for the thick filament, nebulin is the measuring tape for the thin filament. It runs the length of the actin and determines how long the actin is allowed to grow. Titin measures the springs on the thick slide. Nebulin measures on the thin slide. After reading this description, it might be a bit confusing for many of our students. That's why I'm going to show a diagram to address this issue. So now, as we have completed the full picture, we can understand what Z line is and what M line is. These are the core components, along with actin and myosin. Then we can add the molecular spring, which we set as titin. You see here signified or symbolized as a spring. And this titin runs from M line to Z line, which provides elasticity. Then we have put a measuring tape, which is called nebulin. And this nebulin determines the length of the actin filament. Let me repeat it. This nebulin runs the length of the actin and determines how long the actin is allowed to grow. So we can say titin measures and springs on the thick side. Nebulin measures on the thin side. Now the table. And I want you to notice that every entry follows from the sliding idea we just built. The A band contains the full length of thick filament including overlap region, and its behavior during contraction is no change in length. Let me say it again. The A band contains the full length of the thick filament including the overlap region. So this region is the A band, and its behavior during contraction is no change in length. Say that out loud. No change. The A band is defined by the thick filament. The thick filament does not change length. Therefore, the A band does not change length, and it is the single most examined line in this table. A quick memory hook that has saved thousands of students. A band is dark. See, I have capitalized the letter A here. On the other hand, I band is light. The letter A hides in the word dark. The letter I hides in the word light. So we can capitalize the I here as well. So dark A and light I. Under the microscope, the dark band is where the thick filaments are. If you see here, you see the dark bands. And it is because thick protein blocks more light. And the I band contains thin filaments only. No thick filaments overlap here. You see here, this is the I band, and it shortens. Of course it does. The I band is the leftover region near the fence post where the actin has not yet met any myosin. As actin gets dragged inward, the leftover region gets swallowed up. It shortens. And you see the I band is a bit lighter compared to the dark A band. And the H zone is the center of A band you see here. It has only thick filaments. There's no overlap and it shortens, may disappear. This is the gap in the middle where the two sets of thin ropes have not yet reached. As they slide inward from both sides, they close that gap. And if the contraction is strong enough, they close it completely. The H zone can vanish. The I band gets thin, but under normal physiological conditions, it does not vanish. Note that difference carefully because there is a trap sitting on it. And the M line that we have set, the M line holds the midline anchor proteins for thick filaments, and its position is constant. It is the center of the sarcomere. Everything moves toward it. It does not move. And the Z line is the thin filament anchor that you see here. And the Z line defines sarcomere boundary. And during contraction, Z lines move closer together. This is the actual definition of the sarcomere shortening. The distance between Z lines is the sarcomere length. Bring the fence posts closer and you have contracted. Let me match the description again because it's very important. We have said A band is the full length of thick filament including overlap region. There's no change in length. I band contains only thin filaments. It shortens. Same for H zone. It shortens. And H zone contains, or the contents of H zone are center of A band and it has thick filaments only, no overlap. M line is the midline anchor proteins for thick filament. Or you can say M line contains the midline anchor proteins for thick filaments and the position is always constant. Z line contains thin filament anchor and it defines sarcomere boundary. And Z line moves closer together. So we have seen it in our image and we have revised it again. And if you find it a bit difficult to understand in the first try, then you can rewind and watch the segment again, because watching it quite a few times can unlock the potential that you feel like that you're not being able to achieve. Now the traps and pearls to wrap up this slide. First one, if the sarcomere shortens, students assume the dark A band shortens too. It doesn't. The A band equals thick filament length and filaments never change length. Only overlap changes. This trap is beautifully designed and I respect. The examiner asks, during contraction, which of the following does not change in length? Then they offer you the I band, the H zone, the A band, and the distance between Z lines and the sarcomere. 4 of those 5 genuinely do shorten. Only one doesn't. And the student who thinks contraction means everything scrunches up has no basis for choosing. So they guess and they lose 0.4. If you take one thing from this slide, take this. A band is the immovable object in a sarcomere. Everything else moves. The A band doesn't. Then comes the second trap. At maximal contraction, thin filaments overlap the entire thick filament, eliminating the H zone. The I band narrows but does not normally disappear. Know which one vanishes. This is the sequel trap. Once you have learned that the A band is constant, the examiner moves to the goalpost and asks which region disappears entirely at maximum contraction. Now you need a finer distinction. The H zone that we have set, you see here, the H zone. This H zone is a gap and the gaps can be closed completely. The I band is a region of actin near the fence post you see here. And there is always some actin sitting outside the reach of the myosin. So it narrows without vanishing. Gap closes fully. Leftover region merely shrinks. And here's the whole slide compressed into one sentence. Filament lengths stay constant. The degree of overlap changes. Thin filaments slide past thick filaments, pulling Z lines toward the M line. If you remember nothing else from the last 15 or 20 minutes, remember your interlaced fingers. Same fingers, closer hands. That is contraction. Every band question on the IMAT is answerable from that one image. And now we can go and find out how the myosin actually does the pulling. We arrived at slide 4 and we have a problem to solve here. And I want you to feel the problem before I give you the answer. In last slide, we established that myosin had reached out and grabbed actin here. Fine, but actin is sitting there permanently and myosin is sitting permanently too. And ATP is floating around your muscle cells all the time. So what stops your biceps from contracting right now continuously forever? What is stopping every muscle in your body from locking onto permanent spasm? There has to be a lock. Something has to physically prevent myosin from touching actin until permission is granted. Now we have our canvas again and we have drawn the actin filament, but this time we have drawn it properly. We see two strands, strand 1 and strand 2. Both of the strands are made up with actin beads, and the strands are twisted around each other in a double helix, like two strings of pearls twisted together. Where two twisted strings meet, you get a long spiral groove. You will see a groove here and also in the other strand. But the groove has been covered by tropomyosin. That's why it's not being visible here. So, to complete our narration, we can say again that where the strands have met, you will get a long spiral groove running down the length. And we have laid a long thin rod into that groove, and that rod indicated here in orange is called tropomyosin. And notice what it does simply by sitting there. It lies directly on the top of the exact spots where myosin wants to bind. That means it has occupied the myosin binding site. It is not doing anything clever. It is just in the way. A body lying across a doorway. But a rod, that means this tropomyosin, could easily roll out. Something has to hold it in position. So every 40 nanometers or so along the rod, we clamp on a small three-part complex you see here. That is troponin, and the word complex matters. It is not one protein. It is three proteins, 1, 2, 3. These three proteins working as a unit. Let me give you the analogy I find sticks best. Think of a door with a security chain. Tropomyosin is the chain across the door. This is tropomyosin you can symbolize as. And troponin is the fitting that holds the chain. Remember the three protein complex? That means this troponin would have three parts. One part is screwed into the chain itself. Here, one part is the piece that keeps the tension on the chain. Here, holding it taut across the doorway. And one part is the lock cylinder, the yellow one, waiting for a key. And the key here, this key is the calcium. Stop considering this as myosin head, rather consider it as calcium. When calcium arrives and slots into the lock cylinder, the whole fitting changes shape. The part holding tension lets go. The chain slackens and rolls deeper into the groove out of the doorway. And suddenly the binding sides that were covered a millisecond ago are wide open. Myosin, which has been ready and waiting the whole time, this myosin will immediately grab on to the binding sides of actin indicated here. Now let us see that again in this diagram. The first one is the relaxed state, and when calcium came, calcium binds to troponin C, and as a result the tropomyosin moves, which exposes the myosin binding sites in actin, and myosin head binds to actin, which creates a power stroke, and as a result the filament starts moving. This arrow indicates the filament is moving, and if the movement is continuous and unregulated, it would lead to permanent spasm. That means the muscle will stay permanently contracted, which we can call tetany as well. Now here's the wrong belief, and it's a subtle one. Students learn that calcium causes contraction, and they picture calcium doing something to the myosin. They imagine calcium activating the motor, switching on the engine, energizing the head. It does none of those things. The myosin head was already energized. We can consider this part as the head of myosin. It was already cocked and ready. Calcium did not start the engine. Calcium got to the obstacle, and it got the obstacle out of the way. And the distinction is worth real marks because it explains something otherwise confusing. Why muscle relaxation requires energy? If calcium were switching the motor on, then removing calcium would just switch it off and relaxation would be free. But calcium is removing a block. So to relax, you have to actively drag every calcium that were here back out of the cell interior and store it away. And that caused ATP. We will come back to that in slide 7. For now, calcium removes a block. It does not power a motor. That we need to know. We are done with the canvas phase. Now we can pivot to our anchor phase. First off, regulatory proteins are thin filament associated proteins that gate myosin access to actin. The word gate is doing all the work in that sentence. A gate does not push anything. A gate decides whether something is allowed through. And note the phrase, thin filament associated. Because in slide 11, we will meet a muscle where the regulation sits on the thick filament instead, and that contrast is examinable. And just like we have mentioned in our canvas phase, tropomyosin is a rod-like coil protein lying in the grooves of the actin double helix, our chain in the doorway. Coiled coil simply means two protein helices wound around each other to make a stiff rod. Imagine like this, which is exactly what you want if your job is to lie rigidly along a groove. At rest, physically occludes the myosin binding sites on actin. Occludes means block. It is a physical obstruction, not a chemical inhibition. Nothing is switched off. Something is simply covered. So, as this was the actin helix that we drew, we can put tropomyosin, let's say, in a different color like this. Let's say it's lying on the groove. That means it has covered or occluded the binding site. And when activated, on activation, it shifts laterally, exposing the binding sites. That means it rolls sideways into the groove. It does not detach. It does not degrade. It does not leave. It just moves out of the way, and it will roll straight back the moment calcium leaves. Now, if we go to key features of troponin complex, let's bring that image so that we don't just guess everything in this slide. As mentioned, this part is the troponin complex. It has three subunits: troponin I, troponin T, and troponin C. The first one, troponin I, is the inhibitory subunit, and it holds tropomyosin in the blocking position. I stands for inhibitory. This is the part keeping tension on the chain. Then troponin T, you see here in purple color. Troponin T is the tropomyosin binding subunit. That means it anchors the complex to tropomyosin. T for tropomyosin. This is the screw that fastens the fitting to the chain. Then comes troponin C. Troponin C is the calcium binding subunit, the regulatory switch. C stands for calcium. This is the lock cylinder waiting for the key. So I for inhibitory, T for tropomyosin, and C for calcium. 3 letters, 3 jobs, and the exam has asked which subunit binds calcium more than once. C for calcium, do not overthink it. Now the sequence or mechanism in order. And the order matters because the exam will scramble it. First, calcium binds troponin C, causing a conformational change in troponin complex. That means the key will come and it enters the lock. And conformational change simply means the protein physically changes shape. Proteins are not rigid. When something binds, they flex. Second, troponin I releases its hold. Tropomyosin rolls deeper into the actin groove. Tension comes off the chain and the chain drops out of the doorway. Third, myosin binding sites on actin become exposed. Cross bridges can form. Note the wording. Cross bridges can form. That means permission has been granted. Formation itself is the next slide. And fourth, running the whole thing backwards. Calcium removed. Troponin I reasserts inhibition. Tropomyosin recovers sites. That means relaxation. Take the key out. That means if you take the calcium out, the lock will re-engage. The chain goes back across the doorway and myosin can no longer bind. Relaxation is not something myosin decides. It is imposed on myosin by the regulatory system. Now the trap alert. First one. In skeletal and cardiac muscle, calcium binds to troponin C. It does not bind directly to actin or the myosin head. This is a frequently offered distractor. Here is how they build the question. They ask, calcium initiates skeletal muscle contraction by binding to which of the following? Options would be actin, then let's say myosin, troponin, tropomyosin, and ATP. Two of those five are extremely tempting. Actin is tempting because calcium exposes actin sites, so students associate the two. Tropomyosin is tempting because tropomyosin is the thing that moves, so students assume calcium must push it. Both wrong. Calcium binds troponin, C, and troponin then moves tropomyosin. There is a middleman, and the middleman is the answer. If it helps, calcium never touches the chain, and it never touches the door. It only ever touches the law. Then exam pearl, we should be careful not to forget. It starts saying threshold is approximately 10 to the minus 6 mole per liter. Below it, no cross bridges form regardless of how much ATP is available. Calcium determines whether contraction occurs at all. That phrase, regardless of how much ATP is available, is the important one. That means here, it tells you that ATP and calcium are not interchangeable and not substitutes for each other. ATP is the fuel. Calcium is the ignition key. A car with a full tank and no key goes nowhere. A car with a key and an empty tank goes nowhere. You need both, and the exam has asked about both failure modes separately. If this lecture is helping you, the full guide goes deeper than I can in a video. Every subject is now covered: biology, chemistry, physics, maths, and logical reasoning and reading. That is the entire syllabus. These are not summaries. Each guide works through what the exam actually tests, how the questions are really worded, and the specific traps that cost people marks every single year. If a mistake shows up in past papers, it is named and taken apart. They're pay what you want, so you set the price. And every guide includes free lifetime updates, which means you buy once and every future version is yours at the same link permanently. Link in the description. Good luck. Slide 5, the cross bridge cycle. And this is the engine room. If you understand this slide properly, 3 or 4 IMAT questions become free every year. So let us build it slowly and get it exactly right. We will start with this diagram. First, imagine one myosin head here, then another one here. If you want to describe it, then how would you describe? You would say that the myosin head looks like a golf club. It has a long shaft coming off the thick filament here and a bulky head at the end here. And that head is a machine with 2 grip sides. One side, let's say this one grabs actin. That's why I've written that actin filament gets attached to it, or actin binding site written here. The other side, the one below, it holds ATP. That's why it is set ATP binding site. And the crucial engineering fact, the one that makes the whole thing work, is that these 2 sides talk to each other. When something happens at the ATP site, that means this one, the actin site responds to. When something happens at the actin site, the ATP site responds. They are mechanically linked. Now, I want to correct a wrong belief before we even start, because if you carry it through this slide, you will get the questions backwards. Almost every student assumes that ATP is what makes myosin grab actin. It feels obvious. ATP is energy. Gripping requires effort, so ATP must power the grip. That is completely inverted. ATP is what makes myosin let go. Let me repeat it. ATP is what makes myosin let go. Let me prove it to you with something you already know, and that is rigor mortis. When a person dies, their muscles do not go floppy and stay floppy. After a few hours, they lock solid, rigid, immovable. You can see this portion to get the concept. It's a rough idea that I've shown, that it has become contracted and it has stayed contracted. Now you can ask yourself, what does a dead body have none of? And the answer would be ATP. It doesn't have any ATP because cellular respiration has stopped. So we have a body with 0 ATP and its muscles are locked in maximum grip. If ATP caused gripping, no ATP would mean no grip and the body would be limp. The exact opposite happens here. That means the body stays in a contracted stage. Therefore, ATP is not the grip, ATP is actually the release. That single piece of reasoning is worth memorizing, not as a fact, but as an argument. Because if you can reconstruct it, you can never get this backwards under pressure. So how does the head get its energy, if not from binding? Here's the trick, and it is genuinely elegant. The myosin head does not use ATP at the moment of the power stroke. It uses ATP in advance and it stores the energy in its own shape. That means ATP gets broken down into ADP and inorganic phosphate, that is PI. We just saw this part in isolated segment, which we will repeat to consolidate our understanding. But before that, think of a mousetrap. That's why we're saying mousetrap analogy here. You use energy to pull the bar back and cock it. You see here, you have pulled the bar and you have cocked it. That energy is now stored in the bent spring. This is the spring. The trap sits there, loaded, doing nothing for hours. Then something touches the trigger, let's say this one, and the stored energy releases in an instant. You see here, the energy is spent long before the snap. That means I wanted to say the energy was spent long before the snap, not after the snap. And myosin works exactly like that. As we have said, it uses the ATP beforehand. The ATP that came here, this ATP splits into ADP and inorganic phosphate, or phosphate you can say, and the energy released does not go into pulling anything. It goes into bending the head backwards into a cocked high energy position. That means you see the head has been bent and the head sits there, loaded, holding ADP and phosphate, waiting. When it finally binds actin, you see here, this is the actin strand and it binds to actin, it releases the phosphate and the trap springs. The head will snap forward and it will drag the actin with it. In the last segment, here, there is some artifact and I want to make sure that you don't get confused while trying to interpret it. So let me draw the actin segment again. This was the actin segment, these are the beads, and this is the myosin head. And when it is snapped, it will keep moving in this direction. That means it will become something like this. But remember, the actin bead will move along with it. So I should draw it like this. This is actin bead here, and it will keep moving in the direction of the myosin head. And that's why I have said that the head of myosin snaps forward and drags the actin with it. So the order of the event is first spend energy, then store it in shape, then release it later on contact, cock the trap, then trigger it. Once you see that, the whole 6-step cycle stops being a list to memorize and becomes a story that could not happen in any other order. Now let's move straight to the anchor slide. The cross-bridge cycle is the repeating molecular sequence converting ATP energy into filament sliding. Repeating here is the key word. This is not a one-shot event. Each head does this 5 or so times per second, over and over, pulling the actin in a little further each time, like a rowing crew pulling a rope hand over hand. Now we move to mechanism, and step 1 is ATP binds to the myosin head. Myosin releases actin, detachment requires ATP. There is our rigor mortis logic in a single line. ATP arrives and the head lets go. Nothing has been hydrolyzed yet. The mere act of ATP docking into its site changes the shape of the actin binding site and the grip fails. Step 2. ATP hydrolyzed to ADP and phosphate. Head cocks into high energy extended position. Products stay bound. The mousetrap that we used as analogy will get loaded. And notice that last phrase, products stay bound. The ADP and the phosphate do not float away, stay sitting in the head. And that is essential because the release of one of them is going to be the trigger later. Step 3, cross bridge forms. Only if calcium has exposed the actin binding site. Here is where slide 4 plugs in. The head is loaded and desperate to bind. But if tropomyosin is still lying across the doorway, nothing happens. The head just hovers there, cocked and useless. Calcium is the gatekeeper for this step and only this step. Step 4. Phosphate released, power stroke. Head pivots about 45 degrees, then filament moves 5 to 10 nanometer. The crab springs. Phosphate leaves, and the head snaps from its cocked position through about 45 degrees, dragging the actin filament along with it. Let's say these are the actin beads. The head was here. When it pivots or slides forward like this, the actin beads will also move forward. And the dragging is 5 to 10 nanometer per stroke. That is an absurdly small distance, but multiply it by hundreds of heads on each thick filament and the thousands of sarcomeres in a row, and you get an arm curling a dumbbell. Step 5. ADP released. Rigor state. Tight low energy binding, no nucleotide bound. The stroke is complete. The ADP leaves, and now the head is bound tightly to actin with nothing in its nucleotide pocket. And that is the rigor state. And I want you to notice what it means. This is a normal, transient, completely healthy part of every cycle. In a living muscle, it lasts a few milliseconds before the next ATP arrives. In a dead muscle, no ATP ever arrives, so every head in the body freezes right there. Rigor mortis is simply the rigor state made permanent. Step 6. New ATP binds. Cycle repeats while calcium and ATP remain available. And by this, we are back to step 1 again. Notice the two conditions in that sentence: calcium and ATP. That means both are required. Remove either and the cycle stops, but as we will see, it stops in two very different states. So let me repeat one more time the whole process. Step 1 was ATP binding to myosin head. Imagine this is myosin head. ATP will bind here. And when ATP comes, the previously bound actin will get detached from myosin head. After that, this ATP will get hydrolyzed into ADP and inorganic phosphate. When this happens, the cross bridge will happen between the head and the actin. After that, this ADP and PI written here, you see, the PI or the phosphate will release and it will create power stroke. That means the head will pivot about 45 degrees, or you can simply imagine that it's sliding forward, which is taking the actin along with it. Then ADP will also release from here, which is the rigor state. And after that, new ATP will come. The ATP that was hydrolyzed, it's not here anymore. A new ATP will come here and it will bind, and the cycle will repeat. I have just roughly explained the whole process here so that you can summarize the whole concept shortly. Now the key features. It's written ATP is required for detachment and for recocking, not for cross-bridge formation. Read that carefully. Two jobs are here for ATP, and formation is not one of them. Binding to actin is spontaneous once the site is exposed. It costs nothing. It is letting go and resetting that cost energy. Then displacement per power stroke, approximately 10 nanometer, which is about the width of a cell membrane. Tiny, but repeated enormously. Myosin head on a thick filament cycle asynchronously, producing smooth continuous force. This is an underappreciated detail, and it answers a question students rarely think to ask. If each head lets go during its cycle, why does the muscle not slip backwards? Because the heads are not synchronized. At any instant, some are bound and pulling while others are detached and resetting. It is a tug-of-war theme where members take turns letting go to regrip, and the rope never slackens because someone is always holding. Asynchronous cycling is what makes contraction smooth rather than jerky. Now let me repeat the trap. After that, ATP is depleted, so myosin cannot release actin, muscles look rigid. This demonstrates that ATP powers cross-bridge detachment, not formation. The exam asks this as a why question, which is unusual and which is why students stumble. Why do muscles stiffen after death? The distractors are always plausible sounding. Calcium floods in from outside. Troponin binds calcium permanently. The pumps go into overdrive. All of those sound like they could cause stiffness, but the actual mechanism is an absence, not a presence. Nothing floods in, something runs out. No ATP, no release. And notice the elegance of the exam design. To answer correctly, you must already have the ATP relationship the right way round. If you think ATP causes gripping, then no ATP should mean no gripping, and rigor mortis becomes impossible to explain. The question is a direct test of whether your model is invert. Second trap. Inorganic phosphate release triggers the conformational change. ADP leaves during stroke, after it has begun. IMAT may test which release event initiates the stroke. This is the finest trap in the lecture, and it separates the well-prepared from the very well-prepared. Both ADP and phosphate leave the head. Both leave around the time of the power stroke. So a student who learned the cycle roughly knows that nucleotides depart during the stroke, but cannot say which one pulls the trigger. The order is first phosphate out, then stroke, then ADP out. Phosphate is the trigger. ADP is a passenger. If you want a hook, P for phosphate, P for power stroke. Phosphate powers the stroke. And now the example which you cannot forget. It says cross-bridge cycling continues only while both calcium and ATP are present. Remove either one and cycling stops, but for opposite reasons. That final phrase, for opposite reasons, is the whole pearl. So let me spell it out because the exam can test either half. Take away calcium and keep ATP. Tropomyosin covers the sides, heads cannot bind, and they detached and stay detached. The muscle goes soft and floppy. That is normal relaxation. Take away ATP now and keep calcium. Heads bind fine, stroke fine, and then cannot let go. They accumulate in the bound state. The muscle goes hard and rigid. That is rigor. Same outcome in the sense that cycling stops. Opposite outcome in the sense that one gives you a limp muscle and the other gives you a locked one. Floppy versus stiff. If a question describes the physical state of the muscle, that description is telling you which molecule went missing. We have built the engine. Now we need to find out who turns the key. And that brings us to neuromuscular junction, or NMJ. On the right, on our canvas, we have the surface of a muscle fiber. When I say muscle fiber, it basically means a muscle cell. So you can use both the words interchangeably. And on the left, we have a nerve fiber coming down from your spinal cord. And right here, we see the junction between the nerve fiber and the muscle fiber surface. That is called neuromuscular junction. Now we will just magnify it and observe it in our next slide. If you look closely, you will see that the fiber and the muscle surface, they never touch each other. There is a gap, a real physical fluid-filled gap, about 50 nanometers wide. Now think about what that gap cost us. The nerve is carrying an electrical signal. Let's say this is the electrical signal that's coming. Electricity travels down that axon at tens of meters per second, and then it arrives at a gap it cannot cross, because a gap full of salty water will not conduct a clean signal. So the body has to do something almost absurd. It has to convert this electrical signal into chemical signal, then throw the chemical across the gap. That means the chemicals will come in this gap and then convert it back into an electrical signal on the other side. That means the electrical signal will come here again. That means first electrical, then chemical, then electrical again. Why on earth would evolution build something so convoluted? Because a gap gives you control. If the nerve and muscle were fused, every signal would pass, always with no possibility of regulation, no possibility of amplification, and no possibility of shutting things down. The gap is not a design flaw. The gap is a control point, and because it is a control point, it is also a vulnerability, which is exactly why so many poisons and one famous disease attack precisely here. That is also why the examiner likes it. Now let us build the machinery. On the nerve side, here the axon ends in a swollen terminal. You see the axon swollen here. This swollen terminal is packed with tiny membrane bubbles. These are the bubbles, and each bubble is stuffed with a chemical messenger called acetylcholine. The green dots represent acetylcholine. You can picture water balloons lined up against a wall, ready to be thrown. You see, these balls are fusing here in the membrane. And here's the part students consistently get wrong, so let me put it up front. The electrical signal arriving at the terminal does not throw the balloons. Something else does. When the action potential depolarizes the terminal, it opens voltage-gated calcium channel, and the calcium rushes into the nerve ending. That means there will be influx of calcium. It is that calcium, not voltage itself, that makes the balloons fuse with the wall and dump their contents into the gap. That means calcium is responsible for making these balloons fuse to the membrane. So calcium is the actual trigger for release, and I want you to file that carefully because we are about to meet a second completely separate calcium event inside the muscle fiber, and the exam will absolutely try to make you conflate them. Acetylcholine floods across the gap you see here in this gap. It takes less than a millisecond because 50 nanometers is nothing for a small molecule diffusing. On the muscle side, this side, the membrane is not flat. It is thrown into deep folds like the pleats of a curtain, and this dramatically increases the surface area facing the gap. And packed into those folds are receptors. You can consider these as receptors. And here is the beautiful bit. These receptors are not signaling proteins that trigger a cascade. They're the ion channel themselves. When acetylcholine binds, the channel physically opens. No second messengers, no delay, no amplification chain. Ligand binds, pore opens. That is why neuromuscular junction is one of the fastest synapses in the body. To see what happens further, let's draw the surface of the muscle fiber again. So this is the muscle fiber. These were the receptors or ion channels. Once they get the signal from acetylcholine, sodium pours in, and as a result, the inside of the cell becomes less negative, which is called depolarization. So we can say that sodium pours in through these open pores, the local membrane depolarizes, and if that depolarizes reaches threshold, an action potential fires along the muscle fiber. And here's a design feature worth noting. At this synapse, it always reaches threshold. The nerve releases far more acetylcholine than strictly necessary, and there are far more receptors than strictly necessary. This is deliberate over-engineering called the safety factor. One nerve impulse produces one muscle impulse every time, guaranteed. Last problem to solve here. Acetylcholine has done its job. If it just sat in the gap, the receptors would stay open and the muscle would stay depolarized forever. So sitting in the gap, anchored to the connective tissue, is an enzyme that chops acetylcholine apart within milliseconds. And the enzyme is acetylcholine esterase. This acetylcholine esterase is the cleanup crew, and it works so fast that a single acetylcholine molecule gets perhaps one shot at a receptor before it is destroyed. Now hold that thought, because it tells you what happens if you break the cleanup crew. If you block this enzyme, and acetylcholine accumulates, receptors would stay open, and muscle goes into continuous, uncontrollable contraction. That is precisely how nerve agents and many insecticides kill, not by poisoning the muscle, but by stopping the off switch. Now we have arrived at the slide of neuromuscular junction after the canvas phase. It starts with the definition. Neuromuscular junction is the chemical synapse between a somatic motor neuron and a skeletal muscle fiber. Two words to notice. First, chemical, because of the gap we just built, and somatic, meaning voluntary nervous system, which tells you this whole slide is about skeletal muscle. Cardiac and smooth muscle are wired by the autonomic system and do not have this classic junction. And now about structure. First one is presynaptic terminal. This presynaptic terminal is the motor neuron axon ending containing acetylcholine vesicles. Our water balloons against a wall that we have just analyzed in the canvas phase. Then synaptic cleft. This is the gap containing acetylcholine esterase anchored to the basement membrane. And motor end plate is the folded postsynaptic sarcomere dense with nicotinic receptors. The pleated curtain packed with receptors. That you need to remember. The mechanism or sequence. 7 steps in strict order, and the exam scrambles them. 1. Action potential arrives at the axon terminal. The electrical signal has traveled from the spinal cord and hit the end of the line. 2. Voltage-gated calcium channels open. Calcium enters the terminal. That means voltage-gated calcium channel opens and calcium comes in from outside. Then 3. Calcium triggers exocytosis of acetylcholine into the synaptic cleft. Exocytosis simply means the vesicle fuses with the membrane and empties outward. The balloons are thrown, if we think about the analogy. 4. Acetylcholine binds nicotinic acetylcholine receptors, which are ligand-gated ion channels. When we say ligand-gated, we mean the chemical itself opens the pore. This is the direct, fast route. 5. Sodium enters and potassium exits. Sodium influx dominates, meaning both ions move because these channels are not fuzzy. They let small cations through. But sodium has a much steeper driving force pushing it inward than potassium has pushing it outward. So the net result is inward positive charge and depolarization. 6 would be end plate potential generated. Always supra-threshold triggers a muscle action potential. That word always is the safety factor we discussed. This is not a maybe. This is must be. 7. Acetylcholine esterase degrades acetylcholine, terminating the signal. That means cleanup, and the muscle is ready for the next impulse. Now the clinical payoff that we can understand. 4 agents, and each one attacks a different step, which is exactly why they make such good exam questions. Curare is a competitive nicotinic receptor antagonist, which causes flaccid paralysis. This is the arrow poison from the Amazon. It sits in the receptor without opening it, so acetylcholine arrives and finds every seat taken. No channel opens, no depolarization, no contraction. The muscle goes limp, which is what flaccid means. And the patient dies of respiratory failure because the diaphragm cannot move. Organophosphates and nerve agents are acetylcholine esterase inhibitors, and they cause sustained spasm. So this is the opposite catastrophe. Break the cleanup enzyme, acetylcholine accumulates, receptors are hammered continuously, and the muscles lock in spasm. Again, the diaphragm fails, but now because it cannot relax rather than because it cannot contract. Notice the symmetry there and how examinable it is. Curare blocks the receptor and gives you floppy paralysis. Nerve agents block the enzyme and give you rigid spasm. Two poisons, one junction, opposite pictures. Then comes botulinum toxin. Botulinum toxin here blocks acetylcholine release from presynaptic terminal. This one attacks earlier still on the nerve side, preventing the vesicles from fusing at all. No acetylcholine is ever released, and this is the same toxin used cosmetically in tiny doses to paralyze small facial muscles. Myasthenia gravis involves autoantibodies against nicotinic receptors and causes fatigable weakness. Here the body attacks its own receptors and destroys them. Fewer receptor means the safety factor is eroded. Early in the day, with vesicle stores full, transmission just about works. As the day goes on and stores deplete, transmission starts failing. That is why the hallmark is weakness that worsens with use and improves with rest. Classically, the eyelids droop by evening. If an IMAT stem describes weakness that gets worse through the day, that word, fatigable, is the diagnosis. Don't forget the trap and pearls. So first, trap says calcium entering the axon terminal triggers acetylcholine release. Calcium release from the sarcoplasmic reticulum triggers contraction. These are two distinct calcium events at two different locations. Okay, let me wrap it up. This is the trap I warned you about during the Canvas build. Students learn calcium causes muscle contraction, then meet calcium at the nerve terminal and merge the two into one event in their heads. They're completely different cell entirely. One is inside the neuron, the other is inside the muscle fiber. Different source. One comes from outside the cell through the plasma membrane, the other comes from an internal store. Different job. One causes vesicles to fuse, the other causes troponin to change shape. The exam exploits this by asking something like, in skeletal muscle contraction, calcium binds to which structure? If you have two events merged, you might reach for something about vesicles. The answer is troponin C, because the question said contraction, and contraction calcium is the sarcoplasmic reticulum calcium. Read the location in the stem and it tells you which calcium is being discussed. Pearl. Transmission at the NMJ is obligatory. Unlike neuron-to-neuron synapses, one nerve action potential always produces one muscle action potential. The end plate potential has a large safety margin above threshold. This is worth understanding rather than memorizing. In the brain, a single incoming signal usually does nothing on its own. A neuron needs many inputs summing together before it fires. That is how the brain computes. But the neuromuscular junction is not computing anything. It is executing an order. The decision was already made upstream in the spinal cord. So the junction is built with massive redundancy to make sure the order gets through every single time. And that also explains myasthenia gravis perfectly. The disease does not create a new problem. It eats away the safety margin until, eventually, some impulses stop getting through. The system was over-engineered, and the disease reveals exactly how much over-engineering there was. We now have to solve the hardest engineering problem in this entire lecture, and I want you to see the problem before you see the solution, because the solution is genuinely clever. In the previous slide, we have seen how the signal was generated, and as a continuation to that, we can say the signal has arrived. An action potential is now sweeping along the surface of the muscle fiber. That's a good thing. But the contractile machinery is not on the surface. The machinery is here, packed solid through the entire volume of the fiber. And that fiber can be 100 micrometers thick. So we have an electrical signal on the outside here, and machinery buried deep inside. Now there can be a question in your mind that why not just let calcium diffuse inward from the surface? Let's say calcium is here, then let it diffuse here. And the answer is diffusion is catastrophically slow over those distances. Calcium diffusing from the surface to the center of a thick fiber would take a significant fraction of a second. Meanwhile, the sarcomeres near the surface would already be contracting. That means this region will start contracting. And as a result, you would get a fiber that squeezes at the edges first and the middle last, tearing inefficiency and useless force. Muscle needs every sarcomere in the fiber to fire at the same instant. So how do you deliver a surface signal to the deep interior instantly? And here is the solution. And it is so simple, it is almost funny. If the signal cannot travel inward, bring the outside inward. Take the surface membrane, you see here, and push it in, tunneling it deep into the fiber like pressing your finger into a balloon, you see here. Now you have a tube running through the interior of the cell whose inside is continuous with the outside world, here and here, and whose membrane is continuous with the surface membrane. And because it is the same membrane and action potential, sweeping the surface simply keeps going. Let's say this is the action potential generated. It will come here, which will go straight down the tunnel at full speed into the core of the fiber. And that is T-tubule or transverse tubule because it runs crosswise through the fiber. Now running alongside these tunnels is the muscle cell version of smooth endoplasmic reticulum, which are called the sarcoplasmic reticulum. Think of it as a network of interconnected bags wrapping around every myofibril. And inside those bags is a huge stockpile of calcium, concentrated thousands of times higher than in the cell fluid. At the points where the sarcoplasmic reticulum or SR runs up against a T-tubule, it swells into enlarged end sacs called terminal cisternae. You see here, pointed. And it presses right up against the tubule wall. One T-tubule in the middle here, this is a T-tubule, one terminal cisterna on each side, one over here, another here. So 3 structures in a row, and that is a triad. Tri for 3. So now we have the electrical signal racing down the tunnel. This is the site of electrical signal. And the stockpile of calcium sitting right beside it. That means inside sarcoplasmic reticulum. And they're separated by 2 membranes and about 12 nanometers of space. The final question is, how does the voltage in the tunnel, that means here, open the calcium gate in the back or sarcoplasmic reticulum? And here is where skeletal muscle does something genuinely remarkable. In the T-tubule, you see the membrane. You can consider this membrane or this membrane. And in this membrane of T-tubule, there is a voltage sensing protein. And in sarcoplasmic reticulum membrane, directly opposite, there is a calcium release channel. And in skeletal muscle, these 2 proteins physically touch each other. The voltage sensor that we described is called the dihydropyridine receptor or DHPR, named after a drug that binds it. The release channel is called the ryanodine receptor or RyR, indicated by the color code red. And this RyR is named after a plant poison. Terrible names that tell you nothing about function, which is why students find them hard. Just remember, DHPR is the sensor on the tunnel, RyR is the gate on the back. When the action potential arrives, you see here, the DHPR changes shape here. And because it is physically attached to the RyR, you see here, and the linkage is indicated here, that shape change is transmitted mechanically like a lever and physically yanks the RyR open. No chemical messenger, no second messenger cascade needed, and also no calcium needed from outside. So you see a protein, DHPR, moves and pulls another protein open. The other protein I'm talking about is this RyR channel, and it is the closest thing in the body to a mechanical linkage. Now, the wrong belief I have to correct, because it is the single most examined distinction in this topic. Students learn about calcium-induced calcium release. That means calcium is releasing more calcium. They learn that calcium entering the cell triggers more calcium release, and they apply it to skeletal muscle. That is cardiac muscle, not skeletal. In cardiac muscle, this DHPR is not just a sensor. It is an actual calcium channel that opens and lets calcium in from outside. You see calcium here getting in. That's why I said that it's a calcium channel that opens and lets calcium in from outside. And that trickle of outside calcium then binds the RyR. This is the RyR and opens it chemically. That means calcium is triggering calcium, which means cardiac muscle absolutely depends on extracellular calcium and skeletal muscle does not. That's why we have written here that this phenomenon depends on extracellular calcium. And you can verify it with a simple experiment. You can take a skeletal muscle and you can put it in a bath. Let's say we have skeletal muscle in this bath and we don't have any calcium, that means zero calcium. And if you stimulate the nerve, then this skeletal muscle will contract repeatedly for a long time because it has its own internal stockpile and never needed outside calcium. Now you can do the same for cardiac muscle. Let's say you take a bath and you put cardiac muscle here and no calcium here as well. And this cardiac muscle will stop contracting within seconds. No outside calcium, no trigger, no release, and that means no beat. Same two proteins and completely different mechanism. Now we are moving straight into our anchor slide. Slide 7 titled T-tubules, triads, and calcium handling. It's not new, but it's required to bolster our concept. So first off, excitation contraction coupling is the process linking sarcolemmal depolarization to calcium release. That is the name for the whole problem we just solved, turning electricity into calcium. And T-tubule is an invagination of the sarcolemma carrying the action potential into the fiber interior. Invagination is the technical word for pushing inward our finger in the balloon that we have just established in our canvas phase. Then triad. A triad is one T-tubule flanked by two terminal cisternae of the sarcoplasmic reticulum. If you consider this is the T-tubule, then on both sides you will have cisternae, one on the left, one on the right. And these cisternae are part of sarcoplasmic reticulum. So we can say one in the middle, that is T-tubule, and two on the sides. That means 3 in total. Now key features. It starts with T-tubules and it says T-tubules ensure synchronous calcium release across all sarcomeres simultaneously. The word synchronous is the entire justification for their existence. That is the problem they solve. Then comes DHPR. This is the voltage sensor on T-tubule membrane. That is, we can say sensor on the tunnel. Then RyR or ryanodine receptor. This is the calcium release channel on the sarcoplasmic reticulum membrane. Then comes resting sarcoplasmic calcium, which is approximately 10 to the minus 7 mole per liter. That means it says 10 to the power minus 7 mole per liter. And peak sarcoplasmic calcium approximately 10 to the minus 5 mole per liter, which is 100-fold rise. Look at those numbers. 100-fold jump in milliseconds. And remember the threshold from slide 4 was 10 to the minus 6, sitting neatly between the two. Resting level is 10 times below threshold. Peak level is 10 times above it. That is not accidental. It gives the system a clean, decisive on and off with no ambiguity in between. This table is the highest yield object in the lecture, 5 rows, and I want every one of them in your memory. First one, DHPR function. In skeletal muscle, it is a voltage sensor only, no ion flow. In cardiac, it is an L-type calcium channel. Calcium enters. Same protein family, two completely different jobs. In one, it is a lever. In the other, it is a doorway. Calcium source. Skeletal muscle uses the sarcoplasmic reticulum only. Cardiac uses extracellular calcium plus SR. This is the row that the zero calcium bath experiment tests. Ranitidine receptor, RyR1 in skeletal, RyR2 in cardiac. Different isoforms of the same channel family, 1 and 2, skeletal first, cardiac second. Coupling mechanism, direct mechanical coupling in skeletal, calcium-induced calcium release in cardiac. Liver versus chemical trigger. And extracellular calcium needed? No for skeletal, yes, essential for cardiac. That single row has clinical consequences. Drugs that block L-type calcium channel, or the drugs that are called the calcium channel blockers used for high blood pressure, reduce the force of cardiac contraction because they cut off the trigger calcium. They do not paralyze your skeletal muscles because skeletal muscle never needed that calcium in the first place. One drug class, one tissue affected, and the reason is sitting in this table. Now the relaxation of the muscle, and one name comes first, that is SARCA. SARCA is the sarcoplasmic reticulum calcium ATPase, and it pumps calcium back into sarcoplasmic reticulum lumen. Note the name ends in ATPase, and that ending always tells you the same thing. This protein hydrolyzes ATP to do its job. It requires ATP, and as a result, sarcoplasmic calcium falls. Troponin C loses calcium. Tropomyosin reblocks actin. That is the full relaxation chain running backwards through everything we built in slide 4. Then calsequestrin. This is a calcium binding protein inside the sarcoplasmic reticulum lumen, which buffers stored calcium. This one is clever and worth 30 seconds of our time. If you try to store that much free calcium inside sarcoplasmic reticulum lumen, let's say this is the sarcoplasmic reticulum lumen. If you try to store that much free calcium, the concentration gradient would become so steep that SARCA could not pump against it. That means SARCA will struggle. So the concentration gradient must be kept in check. And calsequestrin sits inside the back or the sarcoplasmic reticulum and binds calcium loosely, keeping the free concentration low while the total stored amount stays enormous. That means the calsequestrin, let's say CQ, it will bind calcium together. So they are bound calcium. And you can imagine that it is like storing water as ice. Same quantity or less pressure. That is what buffer means here. Now coming to the traps. First one, it says, in skeletal muscle, the DHPR does not conduct calcium. It physically pulls open RyR1. In cardiac muscle, the DHPR is an L-type calcium channel whose calcium influx triggers RyR2. This is the single most tested distinction in EC coupling. The exam question usually reads, in skeletal muscle, what is the immediate consequence of DHPR activation? And then it dangles calcium influx from the extracellular space as an option. That option is describing cardiac muscle, planted in a skeletal muscle question. It is a pure tissue swap trap. One phrase to lock in. In skeletal muscle, the DHPR is a hand, and in cardiac muscle, the DHPR is a door. A hand pulls something open, a door lets something through. Second trap is about relaxation. It says relaxation is not passive. SARCA consumes ATP to resequester calcium. This is why ATP depletion impairs relaxation as well as contraction, and why rigor is a stiff rather than a floppy state. Students find this genuinely counterintuitive. Relaxation feels like the absence of effort. You stop trying, the muscle goes off, or the muscle goes soft. But at the molecular level, relaxing is an uphill fight. You have to grab every calcium ion out of the fluid and push it back into a compartment where the concentration is already thousands of times higher. That is pumping against a gradient, and pumping against a gradient always will cost ATP. And notice how neatly this ties back to rigor mortis. In dying muscle, ATP falls, so SARCA will stop. And as a result, calcium will stay high. So tropomyosin stays open and simultaneously myosin cannot detach. Two failures compounding, both from the same missing molecule, and the result is a muscle locked harder than lying limp. The example talks about 3 places where ATP is consumed in muscle, and I want you to be able to list them cold. First one is cross-bridging cycling, then comes Sarcomere-mediated calcium reuptake, and the sodium-potassium ATPase, restoring ion gradients. A question asking where muscle uses ATP has all 3 as valid targets. Most students can name the first one. Fewer name the second. Almost nobody spontaneously names the third. But the sodium-potassium pump is running continuously in every muscle fiber you own, resetting the ion gradients after every single action potential. And it is a major consumer of your resting energy budget. So if a question asks, which process in a muscle fiber does not require ATP, be careful, because the list of things that do require is longer than you think. And the correct answer to that question is usually the action potential itself, which we will get to in slide 14 and which is entirely passive. We now zoom out. For 7 slides, we have been inside a single fiber. Now let us look at the whole muscle and answer a question that sounds simple but is not. You use the same muscle to pick up a feather and also to pick up a suitcase. But we established in slide 5 that a muscle fiber is all or none. It either fires a full action potential and gives a full twitch, or it does nothing. There is no half switch. So if every fiber only has one setting, how do you produce a gentle force? And the answer is that nervous system does not control how hard each fiber pulls. It controls how many fibers are pulling and how often. Now in our canvas, we see one motor neuron leaving the spinal cord. As this motor neuron approaches the muscle, it branches. You see the branches here? For approximation, we can count 1, 2, 3, 4, 5. 5 branches, another one 6 branches. And this branch, each of the branch ends on a different muscle fiber. You see one ended here, another here, and the list goes on. Every fiber that neuron touches is now under its command. Fire that neuron and every one of those fibers contracts together, always, with no possibility of selecting a subset. That team, the neuron plus all its fibers, is a motor unit. It is the smallest thing the nervous system can switch on. Now think about team size. Consider the tiny muscles you see here, such as superior rectus, and this one is inferior rectus. Actually, it's not superior, it's inferior rectus. These are tiny muscles. And these tiny muscles move your eyeball. If those were controlled in big blocks, your gaze would jump around in coarse jumps and you could never track a line of text. So each neuron there commands only a handful of fibers. You see, some fibers here, some fibers here. That's why I say they command only a handful of fibers. Tiny teams, exquisitely fine control. Now consider the muscles holding you upright, seen here. Precision is irrelevant here. What matters is bulk force, sustained for hours. So each neuron there commands thousands or 2,000 fibers. You see, all are activated. Huge teams, coarse but powerful. That's why it says high force. So we can tell precision equals small teams. Power equals big teams. And you can predict which any muscle has just by asking what it is for. Now the second question is, in what order do you switch the teams on? Suppose you always recruited the biggest teams first. Then the smallest force you could ever produce would be enormous, and picking up an egg would be impossible. So the order must run the other way. Small teams first, then comes the big teams. That means we can say small teams first and big teams last. And that is exactly what happens, and it is not a conscious decision. It falls out of the physics. Small motor neurons have smaller cell bodies, higher electrical resistance, and therefore reach threshold at lower input. They fire first automatically. The nervous system does not choose the order. The order is built into the wiring. That is the size principle. One more layer. recruiting more teams is not the only lever. By team, I mean units. What you can do is you can also fire the same unit or team faster. Here is the analogy I like. Imagine pushing a child on a swing. If you give one push and the swing rises, and then after rising it falls back again, and that is we can call a twitch. Now push again before it has fully returned. You see here that we are pushing before it fully returns, and this time the swing goes higher than the first time in the second push you see. And if you keep pushing it at the right rhythm, the swing climbs and climbs. That means the swing will go higher than before. Nothing about your individual push changed. What changed is that you stopped waiting for the system to reset. The whole analogy can be called summation. And the reason it works in muscle is calcium. One action potential dumps calcium. That means action potential is generated and calcium is dumped. And after that, Sarca, the pump responsible for moving the calcium inside the sarcoplasmic reticulum, works to pump back the calcium. But if a second action potential arrives before Sarca has finished the job, the new calcium lands on top of leftover calcium. That means that there were already some calciums here, and if you push another action potential, let's say the second one, there will be more calcium, and Sarca has a limit. Sarca cannot pull or put back all the calcium at once. That means there will be more calcium and more troponin would be activated and more cross bridges cycling will be there, which equals more force. Push the frequency high enough, you see here, and the Sarca never gets a chance to catch up at all. Calcium simply stays high permanently, that you see here. Every troponin is occupied, every available cross bridge is cycling, and the force stops oscillating and locks in at maximum. And that is called tetanus, you see in the graph, and it is not a pathological state. It is how you hold a heavy bag. Every strong sustained contraction you have ever made was tetanic. So let's look at the graphs again. If you give only one push, that means one action potential will create a twitch. If you keep pushing, then the twitches would be stronger than before. Those are summation. But if you keep giving high frequency constantly, then there won't be any chance of Sarca pumping back the calcium into the sarcoplasmic reticulum. And the result will be tetanus, which is maximum sustained force. Remember, this is not any pathological state. This is a physiological state that your body does when it needs to have a sustained maximum force. To match our anchor slide with canvas face, let us read the definition, key features, size principles, mechanism, and we will follow that with pearls and traps. Okay, first, motor unit. A motor unit is one motor neuron. Together with all the muscle fibers, it innervates. That means neuron plus its team. The team that we were referring to were fibers. And note the word all. You cannot fire half a motor unit. What about the key features? First, it says all fibers within one motor unit are the same fiber type and contract simultaneously. That is an important detail we will use in next slide. A motor unit is metabolically uniform. It is entirely slow fibers or entirely fast fiber, never a mixture. That's why it says same fiber type. Then small units, 2 to 10 fibers, as in extraocular muscles, allow fine precision. It's talking about our eye muscles because our eye movement needs precision. Then it can have large units, which are 1,000 to 2,000 fibers, seen in postural muscles, and it favors bulk force. That means it supports our back and leg by back and leg muscles. Then innervation ratio, fibers per motor neuron, low for precision, high for power. That is the technical term for team size. Low ratio, fine control, and the ratio, if it is high, then there will be brute force, or we can generate brute force with it. If we look at the principle, what it says, motor units recruited in order from smallest to largest. That means smallest would be first, largest would be last. And specifically, Type 1 recruited first, then type 2A, then type 2X only at maximum effort. Notice that last phrase, only at maximum effort. This has a real consequence that athletes exploit. If you never train near maximum intensity, your type 2X units are barely get recruited at all because the size principle never reaches them. That is the physiological reason why heavy or explosive training is required to develop them and why light repetitive work does not help in this regard. And we have also said that single twitch means one action potential, brief force rise and fall lasting 10 to 100 milliseconds. That can be matched with one push of the swing. Then web summation. This is the second stimulus before full relaxation. Force builds on residual calcium. Note the phrase residual calcium. That is the mechanism in 2 words. The leftover calcium from the previous stimulus is what allows the next one to build higher. Unfused tetanus. Partial fusion. Force oscillates at an elevated level. The swing is climbing but still visibly swinging. On a force tracing, you see a wobbly line trending upward. Then fused tetanus. It says circa overwhelmed. Calcium stays saturating maximum sustained force. Okay, so the wobbles disappear and the line goes flat at the top. Circa has lost the race completely. It's just mentioning about the graph here that it has become flat. Then recruitment. Additional motor units activated to increase whole muscle force. And this is the second lever. Frequency gives you more force per unit. Recruitment gives you more units. The nervous system uses both together continuously without you ever thinking about it. And this trap catches students off guard. The trap will catch students who learned all or none as a slogan without asking what unit it applies to. Let us read the trap before we explain. It says that a single fiber gives a full twitch or nothing. The whole muscle produces graded force because different numbers of motor units are recruited. Applying all or none to the whole muscle is the error being tested. That's why I started by saying that this trap catches students who learned all or none as a slogan without asking what unit it applies to. The exam might state that a whole skeletal muscle obeys the all or none law. That statement is false. Or it might ask how a muscle produces graded force despite the all or none law, and offer distractors like fibers contracting partially or calcium being released in variable amounts within a fiber. Both are wrong. The gradation happens above the level of the fiber, not within it. And one line to carry. The fiber has one setting. A muscle has a volume knob, and the knob is the number of units switched on. Now we have come to the pearl, and the title is Why Tetanus Produces Maximum Force. And let us read before we explain it. It says, at high stimulus frequency, Circa cannot re-sequester calcium between action potentials. Sarcoplasmic calcium stays saturating, troponin remains fully activated, and every available cross-bridge cycles continuously. I want you to appreciate what this pearl is really saying, because it explains why a single twitch is actually a weak and an efficient event. In a single twitch, calcium floods in and Circa immediately starts clearing it. Let's say this is the Circa. The calcium, it will throw it off and it will put into sarcoplasmic reticulum again. So calcium is only above threshold for a brief window. Many cross bridges never get the chance to attach before the sides close again. You are not getting the muscle at full capacity. In tetanus, the calcium window never closes. Every cross bridge gets its chance. That is why tetanic force is roughly 3 to 5 times greater than a single twitch in the same muscle, and why physiologists use tetanic force as the reference for 100%. And file this away for slide 12 because it sets up one of the best questions in the lecture, that your heart can never do the tetanus state, ever. And the reason it cannot is what keeps you alive. And if you want to test yourself properly, the biology question bank is at myelinhq.gumroad.com. along with all 5 subject rights. Now we want to understand the concept of different muscle fibers. First off, you can look at a chicken. The breast meat is white, the leg meat is dark. Same bird, same species, same basic muscle protein, but completely different color. And why is that? Because a chicken walks around all day and flies almost never. The legs work continuously for hours, so they are built for endurance. The breast is used for a violent burst of flapping to escape a predator, then nothing for the rest of the day. So it is built for explosive power. Now, what does endurance actually require mechanically? It will require oxygen, because only oxygen-based metabolism can produce ATP indefinitely. To use oxygen, you need lots of mitochondria, because that is where oxygen-based ATP production happens. You need lots of capillaries to deliver oxygen as well. And you need something inside the muscle to hold onto oxygen and hand it to the mitochondria on demand, which is the myoglobin. Myoglobin is a red pigment closely related to hemoglobin. So endurance muscle is packed with myoglobin, and myoglobin is red. And this color is not decoration. The color is a direct readout of the metabolic strategy. Dark meat is dark because it is built to breathe. And what about breast muscle? This breast muscle takes the opposite path. It does not need to sustain anything, so it skips the mitochondria, skips the dense capillary bed. That means fewer capillaries. It also skips the myoglobin, that means low myoglobin, and instead stockpiles glycogen for rapid anaerobic breakdown. No myoglobin, no red. That's why white meat. Now here's the wrong belief that catches students, and it is a subtle one about the word ATPase. Students reason like this. They say type 1 fibers are the endurance fibers, and endurance means good at using energy. Therefore, type 1 fibers must have high enzyme activity. It sounds completely sensible, but it is exactly backward. The enzyme myosin ATPase is the enzyme in the myosin head that splits ATP during cross-bridge cycle. How fast it works determines how fast the head cycles, and how fast the head cycles determines how fast the muscle contracts. Now let me tell you a chain. So that means high ATP causes fast cycling, and this fast cycling means fast contraction. And after that fast contraction, we can deduct as fast ATP consumption. And finally, it will lead us to fast fatigue. So high ATP, actually this is not ATP, it will be high ATPase. And this high ATPase activity is the signature of a fast, fatigable fiber, not an enduring one. The type 1 fibers, they contract slowly, precisely, because their myosin ATPase will be low. Their endurance comes from their mitochondria and blood supply. You see here, abundant mitochondria and the blood supply, which is a completely separate property from the speed of the motor itself. And separate those two ideas, how fast the motor runs and how good the fuel supply is. Type 1. This type 1 has a slow motor with an excellent fuel supply. If we say type 2X, it has a fast motor with a poor fuel supply. Once you hold them apart, the table becomes obvious. Now the elaborate discussion on canvas phase will consolidate our knowledge on this anchor phase. That means slide 9 muscle fiber types, anchor phase will be discussed with definition, comparison table, traps and pearls. Okay, so first of all, it starts with definition and it says fiber types classified by contraction speed, dominant metabolic pathway and fatigue resistance. 3 axes, and they are not independent. They travel together in predictable packages, which is the whole point of the classification. Now let us read the comparison table across, because the internal logic is what makes it memorable. First, common name. Type 1, slow oxidative, then type 2A, fast oxidative, glycolytic, and type 2X is fast glycolytic. The names literally encode speed plus fuel strategy. Type 2A sits in the middle and uses both routes, which is why it is the most adaptable fiber and the one most changed by training. Then comes contraction speed, slow, fast, very fast. And fatigue resistance runs directly opposite, high, intermediate, and low. Speed and stamina trade off against each other in every row of this table. Then myoglobin, high, appears red. Intermediate, and in case of type 2X, it's low and appears white. Remember chicken leg and chicken breast analogy that we have put earlier? That applies here in the myoglobin content. Then mitochondria. Type 1 has many, type 2 has many, but type 2X has few. And capillary density, high, high, and low. Notice that type 1 and type 2A agree on both rows. That is what the word oxidative in both their names is telling you. Then comes primary ATP source. First one, in case of type 1, is oxidative phosphorylation, then oxidative plus glycolysis, and then comes anaerobic glycolysis. And the full slide will be on this next. Then comes glycogen stores. First one, in case of type 1, is low to moderate. Then comes moderate for type 2A and high for type 2X. This one reverses the pattern, and it should. A fiber running on anaerobic glycolysis needs a large local sugar stockpile because it cannot rely on a rich blood supply it doesn't have. Then myosin ATPase activity, low, high, and very high. There it is in print that we discussed. Type 1 is low. Read that row twice because it is the trap. Then motor unit size, it goes small, intermediate, and large. And this ties straight back to the size principle from last slide. Small units are type 1 and get recruited first. Large units are type 2X and get recruited last. You see here? The two slides interlock perfectly. Then typical activity. For type 1, you see posture and marathon. Then type 2A, middle distance and swimming. And type 2X, sprint and throwing. If a stem describes an athlete, this row tells you which fiber type they are asking about. And as usual, let's discuss the trap and pearl, because these are the places where students lose marks and they can easily gain marks as well. So first trap, it's about type 1 having low myosin ATPase activity. Let's read it out loud. It says, endurance capacity and enzyme activity are different properties. Type 1 fibers are fatigue resistant but have low myosin ATPase, which is precisely why they contract slowly. High ATPase equals fast cycling equals fast fatigue. The exam builds this beautifully. It asks which fiber type predominates in an endurance athlete and why. And among the option, it puts type 1 fibers because they have the highest myosin ATPase activity. The fiber type is correct, but the reason is wrong. And a student who is scanning for the words type 1 will tick it and lose the mark. This is a broader lesson about IMAT technique, so let me say it plainly. When an option has two clauses joined by because, both the clauses, the first and the one that comes after because, both clauses must be true. A correct claim with a false justification is still a wrong answer, and examiners use that structure deliberately. Now the exam pearl to make your marks shine amongst the crowd. Let's read it. It's about color maps to metabolism. So red equals type 1, that means high myoglobin, many mitochondria, oxidative. White equals type 2X, low myoglobin, few mitochondria, glycolytic. Myoglobin stores oxygen, so oxygen-dependent fibers are red. This is a recovery tool in the exam. If you blank on the details, start from the color. Red means oxygen, and oxygen means mitochondria, and mitochondria means endurance. Endurance means slow and fatigue resistance or fatigue resistant. And slow means you can say low myosin ATPase. One visual fact unpacks the entire column. Now we will study slide 10. Before going to the anchor slide, we will get familiar with all the ATP related mechanism and caveat. Okay, so here's a fact that would genuinely alarm you. If you sprinted right now using only ATP, which are already sitting inside your muscles, you would run out of it in about 2 seconds. Let me repeat it, only 2 seconds, because your muscle carries almost no ATP installed. And that sounds like a terrible design until you think about why. ATP is large and highly charged molecule. Storing enough of it to run a marathon would require a quantity that would make your muscles absurdly heavy. We are just imagining that ATP is stored in large quantities and it will cause the muscle to become heavy. And also, this amount of ATP would wreck the osmotic balance of the cell. That means if ATP is in huge amounts inside the cell, then cell will start drawing water toward it, which will just cause it to wreck or get lysed. So, after all these drawbacks, evolution made the opposite choice. Evolution decided that store no ATP at all, or little ATP, and instead built extremely fast machinery to regenerate it continuously. Your muscle is not a fuel tank. It is a refinery running at full speed. But now you have a timing problem. Different activities need ATP delivered at completely different rates and for completely different durations. And no single system can do both. So the body built 3 and they hand over to each other like relay runners. Now let us think about the first 2 seconds of a 100-meter sprint. The gun goes, you need enormous ATP instantly. Oxygen-based metabolism cannot help. It takes many steps and needs oxygen delivery that has not even ramped up yet. Even glycolysis, which is fast, takes an enzymatic step to get going. You need ATP in milliseconds. So the muscle keeps an emergency reserve, but not as ATP. It stores it as phosphocreatine, a creatine molecule holding a high energy phosphate. One enzyme, creatine kinase, you see here. And this creatine kinase transfers that phosphate, this phosphate we're talking, straight into ADP, and you have ATP as a result. One step, no oxygen required, and this is essentially instantaneous. Think of it as a power bank in your pocket. It is not the mains supply. It does not last long, but it works the instant you plug in. And here is the elegant part. Because this reaction is so fast, ATP concentration in a sprinting muscle barely drops at all in the first seconds. Phosphocreatine is not really a fuel so much as a buffer, holding ATP steady while the slower systems spin up. But the power bank is small, and 8 to 10 seconds after it starts, it becomes flat, indicated here in the second graph, where you see that ATP stores are getting depleted. So, system 2 takes over, and that is glycolysis. Break glucose down to pyruvate. You see glucose being broken down into pyruvate. And net 2 ATP per glucose. No oxygen required here. Fast to start and decent output. But here's a catch. Glycolysis needs NAD+ to keep running, and it converts NAD+ into NADH as it goes. Without oxygen, the mitochondria cannot recycle that NADH back. So glycolysis would grind to a halt within seconds, choked by its own product. And the fix is to dump the electrons somewhere else. So you're going to take pyruvate and you reduce it to lactate, and NAD+ is regenerated. Lactate is not the goal. Lactate is the price you pay to keep NAD+, this molecule, available. That reframing matters, and we will come back to it because it demolishes something you were probably taught at school. The system carries you for perhaps 30 seconds to 2 minutes of hard effort, enough for a 400-meter race. And the reason a 400 hurts in a way 100 does not. Then system 3, the mains supply. The pyruvate enters the mitochondria and it becomes acetyl-CoA, runs the citric acid cycle, feeds the electron transport chain, and yields roughly 30 ATP per glucose, 15 times the anaerobic yield. It needs oxygen. It takes a couple of minutes to reach full output, and once running, it is essentially inexhaustible, and if you add fat oxidation to it, you can go for hours. So, three systems. First one is instant and tiny, then fast and moderate, and then slow and enormous. First one is power bank, then portable generator, and the final one is the national grid. After completing the canvas phase, now we can safely move to our anchor phase to match what we have studied. And I'm hopeful that the whole slide will be comprehensive to you right now. So let's start with the definition. It says stored ATP in resting muscle lasts only 1 to 2 seconds of maximal contraction. And that is the alarming fact that we see in print. Then three metabolic systems supply ATP in sequence according to intensity and duration. In sequence. They are not alternatives you choose between. They hand over. Now the comparison table. First, phosphocreatine. Duration 0 to 10 seconds. Oxygen no. And creatine kinase transfer phosphate from PCR to ADP, which is immediate. The power bank analogy that we have used earlier. Then anaerobic glycolysis. Duration is 10 seconds to 2 minutes. No oxygen required, and 2 ATP per glucose is generated. And pyruvate reduced to lactate to regenerate NAD+. Note the reasoning is spelled out on the slide. Lactate exists to regenerate NAD+. Then oxidative phosphorylation. Duration beyond 2 minutes. Oxygen required? Obviously yes. And about 30 ATP per glucose yielded here. Citric acid cycle plus electron transport chain. So we can say 15 times the yield compared to anaerobic glycolysis, and that yield ratio is itself is examinable. Now moving to key features. First of all, phosphocreatine buffers ATP concentration during the first seconds of intense effort. Remember, it's buffer, not fuels. That word is precise, and it is the reason creatine supplements work at all. More stored phosphocreatine means a slightly longer buffer window during repeated maximal efforts. Then fatty acid beta oxidation dominates during prolonged low-intensity exercise. Fat is dense and abundant but slow to mobilize, so it comes into its own long, gentle efforts, not on hard ones. Then fast-twitch type 2X fibers rely on glycolysis. Slow-twitch type 1 relies on oxidation. And there is a link straight back to slide 9 that we studied. The fiber type and the fuel system are two descriptions of the same design decision. Now we will talk about the muscle fatigue mechanisms again. 5 mechanisms, and this list is more modern than most textbooks students have used. First, hydrogen ion accumulation causing acidosis, inhibiting myosin ATPase and troponin sensitivity. So, it means that falling pH interferes with both motor enzyme and the calcium sensor. 2 hits at once. Then inorganic phosphate accumulation, reducing calcium sensitivity and impairing the power stroke. And this one should ring a bell from slide 5. Phosphate release drives the power stroke. If the cell is already full of phosphate, the concentration gradient favoring phosphate release is weakened and the stroke is impaired. The fatigue mechanism is the cross-bridge cycle running in reverse conditions. Then, impaired calcium release from the sarcoplasmic reticulum, reducing tetanic force. So, the RyR channels themselves become less effective during sustained activity. So, less calcium comes out per impulse. Then, glycogen depletion, limiting substrate availability during prolonged exercise. This is what marathon runners call hitting the wall. Not acid, not phosphate, simply running out of stored sugar. Then central fatigue, reduced motor drive from central nervous system. And this one is not in the muscle at all. The brain reduces its output before the muscle is physically incapable, which is thought to be protective. It also means a shouted instruction or a finish line in sight can genuinely increase force, which is not psychology alone, it is central drive to be precise. Don't forget the trap and pearl, because if you understand them and if you can deduce them from options, then it means that you have understood the whole slide clearly. Okay, so the first one is about lactate not being the primary cause of fatigue. So current evidence attributes fatigue to hydrogen ion accumulation, inorganic phosphate buildup, and impaired sarcoplasmic reticulum calcium release. Lactate itself is a usable fuel. Mechanism-based questions target the ionic changes, not lactate. Okay, you're almost certainly thought that lactic acid builds up in muscle and causes burning and soreness. That model is outdated. Lactate and hydrogen ions are produced by related but separable processes, and lactate itself is exported, taken up by the heart and by slow twitch fibers and burned as fuel. It is a currency, not a waste product. Delayed soreness the next day is a microscopic muscle damage and inflammation and has nothing to do with lactate, which has cleared within an hour. Now, a practical warning for the exam. If a question simply asks what accumulates during anaerobic glycolysis, lactate will be perfectly correct answer and you should say so. But if a question asks by what mechanism fatigue impairs contraction, the answer sits in the ionic changes. First would be hydrogen ion, then would be phosphate and calcium release. Read whether the stem is asking what accumulates or how contraction fails. Now the exam pearl. A 100-meter sprint uses phosphocreatine. A 400-meter run uses anaerobic glycolysis. A marathon uses oxidative phosphorylation and fatty acid oxidation. IMAT asked this as a duration matching question. This is one of the easiest marks in the whole topic if you approach it correctly. Do not try to recall which system goes with which sport. Instead, ask one question. How long does the event last? If it's 10 seconds or less, phosphocreatine. If it's under 2 minutes, it will be glycolysis. And longer than that, it will be oxidative phosphorylation. The duration is always given in the stem, and the duration is the answer. Let me pose a design brief and see if you can predict the answer. I need a muscle that will wrap around every artery in your body and squeeze them slightly, continuously for 80 years without stopping. It must never fatigue. It must use almost no energy because it never gets a rest. And speed is completely irrelevant. It has all the time in the world. Now, could you build that out of skeletal muscle? Absolutely not. Skeletal muscle burns ATP furiously while holding force, because every cross bridge is cycling on and off many times per second. Holding a contraction with skeletal muscle is expensive. Your arteries would consume a colossal fraction of your daily energy just staying narrow. And as a result, if we put skeletal muscle cell or skeletal muscle fiber in artery, the design will fail. And to solve that problem, we need to introduce smooth muscle in our design. So smooth muscle is not simply a weaker or slower version of skeletal muscle. It is a different solution to a different problem, and every structural difference follows from that. To build the concept of smooth muscle, first throw away the sarcomeres of skeletal muscle. The precise repeating alignment is what you need for speed and directional pull. Smooth muscle needs neither. So its actin and myosin run in loose diagonal crisscrossing bundles anchored to protein blobs scattered through cell and pinned to the membrane. Those blobs are dense bodies doing the job Z-lines do, but scattered instead of aligned. No alignment, that means no stripes. And this has an unexpected advantage. Because the filaments are not locked into a fixed geometry, smooth muscle can generate force across an enormous range of lengths. Think about your bladder, nearly empty or holding half a liter. A skeletal muscle stretched to that degree would be useless because the filaments would barely overlap. Smooth muscle keeps working. The disordered arrangement is a feature. And now, do you remember troponin of skeletal muscle that sat on the actin filament? You need to throw that away too in case of the smooth muscle. That's why you see a cross given here, that no troponin anymore. And here's the deep point that most students never appreciate. In skeletal muscle, regulation sits on the thin filament. That means actin. If you remember, then you will understand troponin, tropomyosin decide whether actin is available. In smooth muscle, the regulation moves to the thick filament. That means to the myosin. The myosin itself is switched on and off. Different filament regulated, entirely different molecular route. Now the question is, how is myosin switched on? And the answer is by phosphorylation. If you attach a phosphate to the myosin light chain, myosin becomes able to work with actin. You see here illustrated. If you remove it, then it cannot. So now the state of the muscle depends on a tug of war between an enzyme that adds phosphate, which is a kinase, and an enzyme that removes it is a phosphatase. So on left hand side you have the kinase, on the right hand side you have the phosphatase. Phosphatase removes and kinase adds. Calcium still triggers everything, but it now works through a different sensor. Calcium will bind to calmodulin. When calcium binds to calmodulin, you see here, it creates calcium-calmodulin complex. And calmodulin will activate the kinase, and the kinase will phosphorylate myosin and force develop. Calmodulin is doing the job troponin C did, but it is a free-floating protein rather than one bolted to the thin filament. Now the final piece, and it is the cleverest thing in this slide. Do you remember the design brief when I said that if you need to hold the forces for decades using almost no energy, then you need to have some specialization. Now contrast that with the phosphorylated cross-bridge cycle that we see in skeletal muscle. And in this cycle, you will need ATP utilization. But to circumvent this, smooth muscle has a trick. A cross-bridge, you see here, and attach to actin, you see, then get dephosphorylated while still attached. That means even after getting dephosphorylated, the head will stay attached. And when that happens, it detaches extremely slowly. It just stays stuck. To understand this, let me give you an analogy. Picture a crowd holding a rope. In skeletal muscle, you see here, everyone is constantly letting go and re-gripping. You see various hands. So they're re-gripping it and constantly letting it go. And that's why it costs energy. In smooth muscle latch state, everyone grips once and simply holds on. Almost no ATP is consumed and the force persists. That is how your arteries maintain blood pressure all day for almost nothing. Some of you might have more curiosity than this. They want to get into deeper concept, but IMAT won't check that much depth. So we're going to stop here with our basic knowledge and we're going to pivot to our anchor phase. Even after that, if you have any question, then you can let me know. I will try to answer them in as detail as possible. So our slide starts with definition as usual. And it says smooth muscle is non-striated involuntary muscle in the walls of hollow viscera and blood vessels. Hollow viscera means hollow organs such as gut, bladder, uterus, airways, anything that is a tube or a bag. Looking into the key features, you will see that smooth muscle has no organized sarcomeres and therefore no striations. Cause and effect stated directly on the slide. The dense bodies serve as actin attachment points, functionally analogous to Z-lines. Scattered anchors instead of aligned fences that we discussed. The no T-tubules. Tabulae serve as membrane invagination for calcium entry. Smooth muscle cells are small enough that they never needed the tunnel system. Tabulae are little flask-shaped dimples that concentrate calcium channels. Then sarcoplasmic reticulum rudimentary. Extracellular calcium is the major source. So, what it means that small internal store So most of the calcium comes from outside, and that makes smooth muscle like cardiac muscle sensitive to calcium channel blockers, which is exactly why those drugs relax blood vessels and lower blood pressure. Then gap junctions link cells in single unit smooth muscle, allowing syncytial contraction. So in the gut and uterus, cells are electrically joined so a wave spreads across a whole sheet. That is how peristalsis works and how the uterus contracts as one unit in labor. While discussing mechanism, you will find 6 steps in order. First will be calcium enters through voltage-gated or receptor-operated channels or via IP3 from SR. That means 3 possible routes in, which reflects how many different signals can command smooth muscle. One can be nerve, another can be hormone and local chemicals. Then calcium binds calmodulin, forming the calcium-calmodulin complex, our replacement sensor that we discussed. And calcium calmodulin activates myosin light chain kinase or MLCK. Note that calcium calmodulin does not act on the filament, it activates an enzyme. There is an extra layer here that skeletal muscle doesn't have, and it is part of why smooth muscle is slower. Then MLCK phosphorylates the myosin regulatory light chains. When it happens, now the cross bridge can work. Then as a continuation, it says that phosphorylated myosin interacts with actin and producing force. Now myosin light chain phosphatase dephosphorylates myosin, causing relaxation. This is the opposing enzyme of MLCK. And notice that relaxation here is not about removing calcium from a sensor. It is about an enzyme reversing a chemical modification genuinely different from skeletal muscle. And this latch state that we have covered multiple times gives the answer how the smooth muscle stays in a tonic contraction. First, dephosphorylated cross bridges remain attached without cyclin. You see here, myosin has a phosphate group attached, and this is phosphorylated state. That means there's a cross bridge. After dephosphorylation, which is done by MLCP, myosin light chain phosphatase, the myosin becomes dephosphorylated. That means no phosphate here anymore and no ATP recycling here. That means the detachment becomes very slow because the myosin changes the shape a bit, and thus it stays in a latch state, or it stays under a tonic force or tonic contraction. That's why it says sustains tonic force at very low ATP cost, essential for vascular tone. And with that, we can discuss our traps and pearls. Trap talks about smooth muscle regulation. In skeletal and cardiac muscle regulation, we need thin filament, which is based via troponin on actin. In smooth muscle regulation, we have thick filament based via phosphorylation of myosin light chains. The regulated filament differs. Most students get as far as knowing that smooth muscle uses calmodulin instead of troponin. Fewer can say that what it implies structurally. This trap tests the deeper version, which filament carries the regulation. Thin filament in striated muscle, thick filament in smooth muscle. A compact way to hold it: in your biceps, the actin decides. In your arteries, the myosin decides. Now the pearl, which talks about MLCK and MLCP at the same time. So it reads, contractile state depends on the balance between kinase and phosphatase activity. Drugs shifting this balance selectively relax smooth muscle, which is the basis of many antihypertensive and bronchodilator therapy. The word balance here is important. Smooth muscle tone is not simply on or off. It is a continuously adjustable setting determined by which enzyme is currently winning. Push the balance toward the kinase and vessels constrict and airways narrow. Push it toward the phosphatase and the vessels dilate and airways open. That is precisely where a large slice of modern pharmacology operates. Asthma inhalers, for example, blood pressure drugs, angina treatment, and drugs used in labor all work by nudging that balance in one direction or the other. And they can do it without touching your skeletal muscle at all, because your skeletal muscle does not use this pathway. The molecular difference is what makes the drug selective. Your heart has beaten roughly 3 billion times. It has never once taken a break, never once cramped, and never once needed you to think about it. Let us find out what had to be engineered differently to make that possible. Start with the problem that keeps me most impressed. Remember, skeletal muscle can tetanize? We spent slide 8 establishing that tetanus is normal and useful, and how you hold a heavy bag had the concept of tetany underneath it. Now imagine your heart doing that. If a heart is tetanized, that means the heart is squeezed shut and held shut, and it won't be able to refuel. If a left ventricle went into tetanus, that means this ventricle, for even 10 seconds, you would lose consciousness. And if it stayed there, you would die. In fact, this is essentially what ventricular fibrillation is. Chaotic, uncoordinated, non-refilling activity, and it kills within minutes. So cardiac muscle must be physically incapable of tetanus, not discouraged from it. Incapable. How would you build a muscle that cannot possibly summate? For that, you need to recall why summation happens. Recall why summation happens. When a second stimulus arrives while the muscle is still contracting from the first, the effect will add. So to make summation impossible, you need the muscle to be unresponsive to any second stimulus for the entire duration of the contraction. In other words, the refractory period must last as long as the contraction itself. In skeletal muscle, you see above, the action potential seen here lasts 1 or 2 milliseconds while the twitch lasts 100. So there are 98 milliseconds of vulnerable window in which a second stimulus can pile on. That means you have this whole region exposed for another action potential. Cardiac muscle closes that window by stretching the action potential out until it matches the contraction. You see that the contraction duration is 200 to 400 milliseconds, and the action potential is also 200 to 400 milliseconds. By the time the heart can respond again, it has already relaxed and refilled. Summation is not forbidden by a rule, it is impossible by timing. How do you stretch an action potential that long? You cannot just leave sodium channels open because they inactivate automatically within milliseconds. So cardiac muscle adds a second inward current that switches on as the sodium current dies, and that is L-type calcium channel. Calcium flows in slowly and steadily inside the cell, and that inward positive charge holds the membrane depolarized against the outward potassium leak. This result is a long flat plateau you see here, and the plateau is calcium. Now the second problem. Skeletal muscle waits for orders from a nerve. If your heart did that, a single injury to one nerve would stop. That means heart cannot wait for the signals from nerve. We cannot accept it. This is unacceptable. So the heart needs to generate its own rhythm, and heart actually does it. And how does it do it? So certain cells in the sinoatrial node that we have watched in the circulatory system lecture, these cells in sinoatrial node simply refuse to sit still electrically. They have channels that leak inward current at rest, indicated here. So their membrane potential drifts steadily upward until it hits threshold and fires. If you are confused about the firing and threshold, don't worry, we're going to cover that in the following slides. Just follow along. So as we were saying, that the cells of sinoatrial nodes, they have some leaky current, and as a result their membrane potential drifts steadily upward until it hits threshold and fires, then resets and drifts up again. They're not receiving a signal, they are a clock. Those channels were named funny channels because they behave oddly compared to every other channel physiologists knew, and the name stuck. And the third problem is coordination. Skeletal muscle fibers can each do their own thing since the nervous system controls which units fire, but the heart must contract as one. If half your ventricle contracted while the other half relaxed, you would generate no pressure at all, just sloshing you will see. So cardiac cells are wired together directly. Where two cells meet end to end, there are pores connecting their cytoplasm. You see here. And ions flow straight through from one cell into the next. If you depolarize one cell and its neighbors depolarize milliseconds later, and the wave sweeps across the entire muscle mass. Electrically, the heart behaves as though it were one giant cell, which is what syncytium means. But now you have created a mechanical danger. Cells that are electrically fused and contracting hard against each other will tear apart at the joints. So at those same junctions, the heart installs heavy-duty mechanical rivets alongside the electrical pores. That combination, electrical pores plus mechanical rivets in one structure, is the intercalated disc. You see here. This is another figure that depicts the intercalated disc. You see here. And this intercalated disc exists nowhere else in the body. Now we have moved to the anchor phase, and we will study the cardiac muscle specialization. Cardiac muscle is striated involuntary muscle with intrinsic rhythmicity and electrical coupling. 4 properties in one line, and 3 of them are unique combination. Striated but involuntary, rhythmic without input, electrically coupled cell to cell. Then the key features start with gap junction. In this image, you see the green color portions. These are the gap junctions. And there's a protein called connexin 43. Connexin 43 is the building block of gap junctions. And that's why it's written that gap junction with connexin 43 is responsible for ionic continuity, allowing action potential to spread from cell to cell. And when we say ionic continuity, it means that the inside of one cell is chemically continuous with the inside of the next. And desmosomes are mechanical anchorage, preventing cells from separating during contraction. The rivets that we have set, here you go, you can see the desmosomes here. These are the desmosomes. And what is the result of the intercalated disc? Due to intercalated discs, the myocardium behaves as a functional syncytium. Remember, functional, not anatomical. The cells remain separate cells. They simply behave as one. And when we set automaticity, it involves the following components: sinoatrial node, funny current channel, intrinsic rate, and conduction pathway. So first, sinoatrial node. It has pacemaker cells with unstable resting potential. Unstable is the operative word. A normal cell sits still at rest, but these cannot. That funny current. This is mixed inward sodium and potassium current driving spontaneous depolarization, which means the drift upward toward threshold. Then intrinsic rate approximately 60 to 100 beats per minute at rest. And note the word intrinsic here. This is the rate with no nervous input at all. Your actual rate is that intrinsic clock modified by the autonomic system. The vagus nerve slowing it down, sympathetic nerves speeding it up. But it doesn't initiate any signal. Then conduction pathway, SA node, then AV node, bundle of His or Purkinje fibers. Learn that order. AV node deliberately delays the signal for about 1/10 of a second, which is what lets the atria finish emptying before the ventricles squeeze. A delay that looks like inefficiency is actually essential timing. Now in this box, we will study cardiac action potential, and for better understanding, we have divided it into 5 phases, and this phase division is universal. So it starts with phase 0, and the event is rapid depolarization, and the ion movement is fast voltage gated. Sodium channels open, sodium rushes in. If you see in the image, let's say this is a cell and sodium keeps rushing inside the cell, which is identical to skeletal muscle so far. Then phase 1, that is early partial repolarization, and the ion movement is transient outward potassium current. That means you will see a brief dip as the sodium channels inactivate and a little potassium escapes from the cell. Then phase 2 is the plateau phase. And in case of ion movement, you will see calcium channels will open. Those are L-type. Calcium enters and triggers CICR. You will see here that L-type calcium channels opened and calciums keep getting inside the cell. You can consider this as a cell. And this is the defining phase and the reason for everything else. Calcium holds the membrane depolarized, and the same calcium is the trigger for calcium-induced calcium release from slide 7 that we have set as CICR. One current doing two jobs at once, prolonging the electrical event and triggering the mechanical one. Phase 3 is final repolarization. Here, delayed rectifier potassium channels open and potassium exits. The calcium channel will close here. Potassium channels will open and membrane falls back. It's very simple to understand. Let's say this is cell and normally the inside of the cell is negative, and if potassium, okay, just make some correction here. Let's say the cell is in this side and the potassium channel has closed. That's why there's no entry of calcium anymore, and potassium is getting outside. Now it's correct. And if that is the case, then the membrane potential will fall back toward negative. And when the membrane becomes negative, we call that repolarization. And if the membrane becomes positive due to the influx of positive ions, we're going to call it depolarization. Then phase 4 is resting potential, stable at negative 85 to negative 95 millivolt in working myocardium. We see in phase 4 here. Note the qualifier working myocardium. The qualifier is working myocardium. In ordinary heart muscle cells, phase 4 is stable. In pacemaker cells, it isn't, and that instability is the funny current we just described. Same phase number, different behavior in different cells, and the exam has used that distinction. That's why you see that stable for working myocardium, but unstable for pacemaker cells. Two graphs given. The flat line represents it's stable, and the line that is showing a slope, it's for pacemaker cells, and it's showing instability. And due to the process, we will see the following consequences. First, action potential lasts 200 to 400 milliseconds, matching the duration of contraction. And matching is the crucial word here. Then long absolute refractory period prevents tetanus in cardiac muscle. The design goal has been achieved by this. Then Frank-Starling mechanism. Greater stretch produces greater force, matching output to venous return. If you want to see the detailed explanation, you can look at our circulatory system for it. Even after that, let us explain, because this one deserves a moment. It means the heart automatically pumps out whatever comes in. More blood returns, the entire ventricle stretches more, the next contraction is stronger, and the extra blood is ejected. No nerve involved here, no brain involved. It is a self-balancing property of the muscle itself, and it is what stops blood accumulating in your lungs every time you stand up or exercise. We will complete this slide with trap and pearls. The trap talks about cardiac muscle not being able to tetanize. Okay, so let's read. The refractory period lasts as long as the contraction itself, so a second stimulus cannot summate. This is physiologically essential, and a tetanused heart could not fill and would fail to pump. The exam asks this in two directions. Sometimes it asks why cardiac muscle cannot undergo tetanus, and the answer is long refractory period. Sometimes it asks what the functional significance of the long refractory period is, and the answer is preventing tetanus so the heart can refill. Same fact asked from cause or from consequence, be ready for both. And watch for the distractor that says cardiac muscle cannot tetanize because it lacks a sarcoplasmic reticulum or because it lacks troponin. Both are false. Cardiac muscle has both. The reason is timing, not missing machinery. Then the pearl, which talks about the plateau phase. And phase 2 is the plateau phase, and phase 2 is sustained calcium influx through L-type channels. This single feature explains the long action potential, the long refractory period, and the impossibility of cardiac tetanus. I want you to see how much this one pearl carries. The plateau is calcium. Because it is calcium, the action potential is long. Because the action potential is long, the refractory period is long. And because the refractory period is long, tetanus is impossible. And because tetanus is impossible, the heart refills. And because your heart refills, you are alive. 5 consequences from 1 calcium ion. If you can reconstruct the chain, you can answer any cardiac action potential question the IMAT writes, including the ones that offer prolonged potassium influx as the cause of the plateau. Potassium moving outward causes repolarization. It cannot possibly hold a depolarization. The plateau must be an inward positive current, and only candidate here is calcium for it. We have spent this whole lecture talking about action potentials arriving and depolarization happening. Now we go underneath all of that and ask what the electrical baseline actually is and where it comes from. This is the foundation, and it also transfers directly to every nervous system question you will ever answer. So we start in our canvas with a cell, and let's say this is cell membrane, and this is the interior of the cell, and this is the outside of the cell. On the inside, you see more potassium, let's put some more, compared to sodium. And on the outside, you will have more sodium. Those are the 2 facts to hold. Potassium lives inside and sodium lives outside. Now here is the crucial question, and almost every student gets it wrong on the first attempt. Given those gradients, why is the inside of the cell negative? The instinctive answer is because sodium is outside and sodium is positive, so all the positive charge is outside. That sounds reasonable, and it is not the mechanism. Sodium is outside, but sodium cannot get in at rest anyway. So it is not doing anything. It is a locked door. The real answer involves potassium leaving, and I want to build it carefully because it is genuinely subtle. At rest, the membrane is riddled with potassium leak channels and almost no open sodium channels. So potassium, let's say this channel, this is potassium channel, this one actually, and through this channel, potassium can leak whenever it wants. And when potassium leaves, it leaves behind a negative charge on the large proteins and phosphate inside the cell that cannot follow. So the inside becomes progressively more negative. I'm putting negative signs here to indicate that. And now something beautiful happens. The growing negative charge starts pulling potassium back in. That means the negative charge will try to pull potassium inside now, because we know opposite charges attract. So potassium has 2 forces acting on it at once and in opposite directions. Concentration pushing the potassium out from the cell, and electrical attraction pulling it in. And potassium keeps leaking out until those 2 forces are exactly equal and opposite. At that point, net movement will stop. Not because potassium ran out and not because the channels closed, but because the outward push and the inward pull are balanced. The voltage at which that balance occurs is around minus 90 millivolts, and that is essentially the resting potential, let's say RP, or sometimes we can tell RMP, that means resting membrane potential. And now here's the wrong belief I have to correct, and it is extremely widespread. Students say the sodium potassium pump creates the resting potential. It does not. It creates the gradients that make the resting potential possible, which is a different claim. Now let me prove it. Poison the sodium potassium pump right now with a drug. Does the membrane potential collapse instantly? No, it stays almost normal for many minutes because the gradients are already established and it takes a long time for them to run down. The pump is not generating the voltage moment to moment. Potassium leaking through open channels is generating the voltage. The pump is the maintenance crew that rebuilt the gradients long ago and keeps topping them up slowly. Now let me give you an analogy to fix the misconception. Think of a water tower feeding a town. The pump that filled the tower is not what gives you shower, it's pressure. Gravity acting on the stored water gives you pressure. That means For pressure, gravity is responsible. The pump just refills the tower slowly over time. I'm talking about this pump. Turn the pump off and you still have water pressure for a good while. Similarly, gradients are the water tower. Leaky channels plus diffusion are the gravity. The sodium-potassium pump is the refill pump in this regard. So you can connect this analogy with the membrane resting potential that we just have established. So this part has resting written here. And you see here that interior of the cell has more potassium and exterior has more sodium, and there's no leaky sodium channel, only leaky potassium channel. Then this leaky potassium channel tries to pull out potassium from interior of the cell to outside. When potassium keeps going out, the cell interior becomes negative, and as a result, the interior of the cells keep pulling the potassium again, and this tug of war will keep happening up until the balance is reached. Now to match it with the anchor slide, we start with definition, and resting membrane potential is defined as the voltage across sarcolemma of an unstimulated muscle fiber. The word to notice is unstimulated. This is baseline, not an event. And in skeletal muscle, it is -85 to -90 millivolt. That means interior negative relative to exterior. And note that this is more negative than a typical neuron, which sits nearer -70. Muscle fibers are unusually potassium permeable at rest, which pushes them closer to the potassium equilibrium value. Now the ion distribution. Potassium, approximately 140 millimole inside and approximately 4 millimole outside. Roughly 35 times more concentrated inside as steep outward gradient. Sodium, approximately 12 millimole inside and 145 millimole outside. Roughly 12 times more concentrated outside as steep inward gradient, sitting there loaded, waiting for a channel to open. That stored gradient is the entire ammunition supply for the action potential. Then calcium, approximately 0.0001 millimole at rest inside and approximately 1.2 millimole outside. Look at that ratio, more than 10,000-fold. The cell works extraordinarily hard to keep internal calcium almost at zero, and that is precisely what makes calcium such a good signal. When it appears against essentially no background noise. Then after calcium, we talk about chloride. It's approximately 4 millimole inside and approximately 120 millimole outside. Chloride is negative, and in skeletal muscle, it contributes significantly to stabilizing the resting potential. Now we will recap the mechanism. At rest, the membrane is predominantly permeable to potassium via inward rectifier channels. Permeability is the key concept, not concentration alone. Which ion is actually allowed to move? We need to consider that, and that is potassium. Then potassium diffuses outward down its concentration gradient. Step 1 of our build. Then impermeant intracellular anions, mainly proteins, are left behind. The negative charge that cannot follow. That interior negative charge accumulates until electrical force balances chemical force. That means equilibrium exactly as we constructed it. Then sodium potassium ATPase, 3 sodium out, 2 potassium in, 1 ATP consumed per cycle. Memorize these numbers: 3 out, 2 in, and 1 ATP. Then the pump maintains the gradients and contributes a small directly electronegative component. Electrogenic. It is not electronegative, it's electrogenic. Electrogenic means it moves net charge because 3 positives leave and only 2 enter. So each cycle removes 1 net positive from the cell. That makes a small direct contribution worth only a few millivolts. Small but real. And examiners occasionally ask about it. We will complete this slide with our trap and pearl. The trap talks about the pump maintaining gradients and the channels creating the potential. Sodium potassium ATPase does not directly generate resting potential. It maintains the gradients that make it possible. The potential itself arises from selective potassium permeability through leak channels. The exam tests this with a drug scenario. It says a drug inhibits the sodium-potassium ATPase and asks for the most likely immediate consequence. If you believe the pump generates the potential, you will predict instant collapse and pick an option saying no action potential can be generated. That is too extreme and too fast. What actually happens is gradual. Sodium slowly accumulates inside. If I draw this cell, sodium will keep accumulating here. And potassium slowly falls. The gradient shallows out and the resting potential drifts less negative. That drift makes the cell more excitable at first and only later causes failure. The correct answer describes a gradual change, not an immediate abolition. Look for the word gradually in options. Now the pearl. It talks about resting potential sits near the potassium equilibrium potential. And it reads, because resting permeability to potassium far exceeds that to sodium, the membrane potential lies close to the potassium equilibrium potential of approximately negative 90 millivolt, pulled slightly positive by a small sodium leak. And here's the general principle hiding inside that pearl, and it is worth more than the pearl itself. The membrane potential always sits near the equilibrium potential of whichever ion the membrane is currently most permeable to. At rest, potassium will dominate, so the potential sits near minus 90. During the peak of an action potential, sodium permeability briefly becomes enormous, so the potential swings toward the sodium equilibrium potential of about plus 60. Nothing mysterious happened. The membrane simply changed which ion it was listening to. If you hold that one sentence, the entire next slide becomes predictable rather than memorized. Now slide 14 will talk about the muscle action potential. We ended the last slide, slide 13, with one sentence that does all the work here. The membrane potential, we write MP for it, sits near the equilibrium potential, let's say EP, of whichever ion the membrane is currently most permeable to. An action potential is nothing more than the membrane rapidly changing its mind about which ion it is listening to, and then changing back. Now let's go to our canvas and look at the diagram. First, focus on the horizontal line at minus 90 millivolt. That is our baseline, and we know why it is here, because potassium is doing the talking. Now the end plate potential from the neuromuscular junction nudges the membrane upward from minus 90 to minus 85, then minus 80, minus 70, minus 60. Nothing dramatic can be seen yet. Then at about minus 55, around here, something snaps. Voltage-gated sodium channels open. If you consider this cell, when the potential reaches negative 55 millivolt, sodium channels will open, and here is the feature that makes an action potential an event rather than a gradual change. Those channels are opened by depolarization. You see here, negative 55, sodium flooding in. And if you open the sodium channels, it will cause more depolarization, which opens more of them. And it is a runaway loop, positive feedback, an avalanche. One pebble dislodges two pebbles, which dislodge four. That is precisely why the response is all or none. You see here. Below threshold, nothing avalanches and the disturbance fades. Above threshold, the avalanche is unstoppable and always goes to completion. There is no such thing as small avalanche. The size of the pebble that started it makes no difference to the size of the landslide. So sodium flooding in, and now the membrane is overwhelmingly permeable to sodium instead of potassium. You see here? So by our rule, the potential races toward the sodium equilibrium potential up past 0 to around plus 30 millivolts here. The inside of the cell is now positive briefly. That means briefly the cell becomes positive. because now the cell is listening to sodium. Now, if nothing stopped this, the cell would be stuck depolarized forever. And to prevent it, two things happen. Two things will stop it. And they are separate mechanisms that students often blur together. First, the sodium channels shut themselves. That's why written automatic inactivation. And this is genuinely clever engineering. So let me be precise. These channels do not simply close. They have two gates. The channels that we're talking about here. And they inactivate. A separate part of the protein swing across and plugs the pore from the inside, kind of like this. A few milliseconds after opening, automatically, regardless of what the voltage is doing. Think of a self-closing door with a timer. It opens when pushed, then slams and latches on its own after a fixed interval, and critically, it cannot be reopened by pushing again until the latch resets. And that latch, you see symbolically here, is the single most important idea on this slide because it explains the refractory period. And the refractory period explains the difference between skeletal and cardiac muscle we met last slide. After that, second, delayed potassium channel open. So you can put potassium channel here, and they sense the same depolarization as the sodium channels, but they respond slowly, arriving late to the party. So this was sodium channel, and this is potassium channel now. And by the time they open, sodium is already inactivating. So now potassium is dominant again. Potassium floods out, and the potential plummets back towards minus. This positive becomes negative, and it plummets toward negative 90 millivolt again. And because those potassium channels are slow to close as well as slow to open, they stay open slightly too long, letting a little extra potassium escape. And the membrane briefly overshoots. You see here in the graph below its resting value down toward minus 100. That is after hyperpolarization, and it is simply the consequence of a sluggish channel. Now the wrong belief I want to dismantle. Students look at all this ion movement and assume it costs enormous energy. They see sodium rushing in. Let's draw the cell again. They see that sodium is rushing in through the channel and potassium is rushing out through the channel, and they think surely that is expensive. Not one molecule of ATP is used during the action potential itself. Every ion moves downhill, down its own electrochemical gradient, through a channel that is simply a hole. These are some simple holes. Channels are not pumps. That means they don't consume energy. The energy was spent earlier by sodium-potassium pump. If we draw here, let's say this is sodium-potassium pump. While it was building the gradients, the action potential just spends the savings and astonishingly little of it. A single action potential moves such a tiny fraction of the total ions that the concentrations barely change. A muscle fiber with its pump completely blocked can still fire many thousands of action potentials before the gradients run down. That is what the water tower analogy was telling you earlier. Jumping straight into our anchor slide and starting with definition, which says action potential is a rapid self-propagating all or none reversal of membrane polarity. Self-propagating because each depolarized patch depolarizes its neighbor, so the signal regenerates itself and never weakens with distance. All or none because of the avalanche. Threshold in skeletal muscle approximately -55 millivolt, the point where the avalanche becomes self-sustaining. The mechanism. First off, end plate potential depolarizes adjacent sarcomere to threshold that we saw on the graph, and it links back to slide 6. Then depolarization. Voltage-gated sodium channels will open, sodium enters, and potential rises to about plus 30 millivolt. The avalanche and the swing toward the sodium equilibrium. Then repolarization. Sodium channels inactivate Delayed rectifier potassium channels open. Potassium exits. Note that both events are listed together because both are needed. Inactivation stops the inward current. That means sodium cannot get inside. Potassium provides the outward current. Neither alone would do it fast enough. After hyperpolarization, slow potassium channel closure overshoots below resting potential, the sluggish channel that we just discussed. Then recovery. Sodium-potassium ATPase restores the ion gradients over subsequent cycles. And notice the phrase over subsequent cycles. Not after each one, not immediately, slowly in the background, exactly as we argued. Now the refractory period. First, absolute refractory period. Sodium channels inactivated, no stimulus of any strength can fire anything. So, start with no stimulus of any strength. That is an absolute statement and it is literally true because the pore is physically plugged. You can apply as much voltage as you like, but the channel, the sodium channel that was there, it is plugged and as a result, no further sodium can get inside. That's why I have said you can apply as much voltage as you like, but a plugged pore does not conduct. Then relative refractory period. Sodium channels recovering here and only a suprathreshold stimulus can fire. It's written. So now some latches have reset, so some channels are available. I mean, in a cell, there is not only one sodium channel. There are more than one. There are numerous. So let's say out of so many sodium channels, some have reset and they are available. But potassium channels are still open, fighting any depolarization. That means potassium channels are open and some sodium channels are reset again. That means this potassium channel is fighting depolarization, and so you need an unusually strong stimulus to win the argument. This is possible but harder. Then skeletal muscle refractory period 1 to 2 milliseconds. So tetanus is possible. And this is short window of unresponsiveness, long twitch, so stimuli can pile up. Then cardiac muscle refractory period 200 to 400 milliseconds, so tetanus is impossible. And there is slide 12, restated from the electrical side. Now coming to the comparison table. First, duration. In case of skeletal muscle, it's 1 to 2 milliseconds. In case of cardiac muscle, 200 to 400 milliseconds. A 200-fold difference from one added current. Then plateau phase. Absent versus present. L-type calcium channels written under cardiac muscle. The cause of the duration difference. Then refractory period. Short and tetanus possible versus long, tetanus impossible. The functional consequence that we discussed earlier. Then resting potential, it's negative 85 to negative 90, and in cardiac muscle, it's negative 85 to negative 95. Essentially similar. This row is the control showing that the differences are about the event, not the baseline. Then depolarizing ion. In case of skeletal muscle, it's sodium, and in cardiac muscle, it's sodium then calcium. Two currents in sequence in the heart. Propagation route. In case of skeletal muscle, this will be along sarcolemma and T-tubules versus gap junctions plus sarcolemma in cardiac muscle. Skeletal muscle spreads the signal within one fiber. Cardiac muscle spreads it between cells. That single row is the difference between a muscle commanded from outside and a muscle that coordinates itself. Now we need to get familiar with the traps and Pearl. The first trap talks about depolarization, meaning less negative. Okay, so read it. Depolarization is any reduction in the negativity of the interior. The brief period when the interior actually becomes positive is the overshoot, a distinct term. Distractors exploit this conflation. This matters because depolarization happens constantly without anything becoming positive. Moving from minus 90 to minus 80 is depolarization. The end plate potential is depolarization. Every subthreshold nudge is depolarization. Only the brief peak of a pull action potential involves an actual positive interior. So if a question asks whether a cell depolarized, Do not look for a positive value. Look for the potential becoming less negative than it was. And in the sodium pump question from last slide, that is exactly the language used. The resting potential becomes less negative. That is depolarization, and it is not positive. Then trap 2 talks about absolute refractory period. It is caused by sodium channels occupying the inactivated state, not by open potassium channels. Open potassium channels account for relative refractory period. Two refractory periods, two different mechanisms, and students routinely give the same explanation for both. Absolute equals, if I write here, absolute equals plugged sodium channel. Nothing can be done. The door is latched. Relative equals open potassium channels. The door works again, but potassium is pushing back, so you need extra force. A hook that works. Absolute is about a door that is broken. Relative is about a door that is heavy. Now example to end this slide. No ATP is consumed during action potential itself. It reads all action potential all ion movement during action potential is passive. Down electrochemical gradients through gated channels. ATP is consumed afterward by the sodium-potassium ATPase to restore the gradients. Okay, so bring this together with the pearl from slide 7 and you have a complete energy map of the muscle fiber, which is exactly what a well-written exam question tests. ATP is consumed by cross-bridge cycling, by sarcomere pumping calcium back into the sarcoplasmic reticulum, and by sodium-potassium pump restoring gradients. ATP is not consumed by action potential and it is not consumed by cross-bridge formation. 4 yeses and 2 nos. If you can sort any process into the right column, you will not be caught by a question asking which of these does not require ATP. This is the last slide before we move to MCQ session. And this slide is different. There is no new mechanism here and no new canvas to build. This is the consolidation pass. Here is how I want you to use it. Do not read along with me passively. Cover the right hand column here. Look at each term and say the hook out loud before I do. Where you can, move on. Where you hesitate, that is a slight number to get back to. 20 minutes of honest self-testing here is worth more than re-watching an hour of lecture. Notice as we go how many of these hooks are traps rather than definitions. That is deliberate. The definitions get you to the right area. The hook gets you past the distractor. Okay, so rapid revision, all key terms. And the table has 3 columns. The term on the left, the one-line definition in the middle, and on the right, the IMAT hook. Starting with sarcomere architecture. Sarcomere is contractile unit between 2 Z lines. Hook 2 to 2.5 micrometers. Filaments slight and never shorten. A-band, full length of thick filament. Hook does not change during contraction. The immovable object. Then I-band, thin filaments only, no overlap. Hook shortens during contraction. H-zone, thick filaments only and center of A-band. Hook can disappear at maximal contraction, the one that vanishes. So for H, you remember it vanishes. Z-line, thin filament anchor, sarcomere boundary, and Z-line move closer together. Titin, elastic protein, M-line to Z-line. Hook, largest protein in the body, and molecular spring. Now regulation, troponin C, calcium binding subunit of troponin. And hook is calcium binds troponin C, not actin or myosin. Tropomyosin blocks myosin binding sites on actin at rest. Hook shifts aside when calcium binds troponin C. Cross-bridge cycle, ATP powered myosin actin cycling. And hook is phosphate release drives the power stroke. Rigor mortis, post-mortem stiffening. No ATP means no detachment. That's why rigor mortis happens. Neuromuscular junction, which talks about signaling, and the definition would be motor neuron to muscle fiber synapse or synapse. Acetylcholine binds nicotinic acetylcholine receptor obligatory 1:1 transmission. Acetylcholinesterase degrades acetylcholine in the synaptic cleft, and inhibition causes sustained spasm. T-tubule, sarcolemmal invagination, and it carries action potential inward for synchronous calcium release. Then triad, 1 T-tubule plus 2 terminal cisternae, and it is site of DHPR and RyR interaction. DHPR, dihydropyridine receptor on T-tubule, and the hook is, in case of skeletal muscle, it's voltage sensor, and cardiac calcium channel. So, and versus door. Then RyR, ryanodine receptor on SR membrane. Hook, RyR1 for skeletal, RyR2 for cardiac. Then CICR, calcium-induced calcium release, and it's cardiac only, needs extracellular calcium. Sarcoplasmic reticulum calcium ATPase, and relaxation requires ATP, not a passive process. Motor unit, 1 motor neuron plus all its fibers, and all or none per unit, not per muscle. Be very careful here. Size principle, recruitment order smallest to largest, and type 1 first, then type 2X last. Tetanus, fused maximal sustained contraction, and calcium stays saturating, impossible in cardiac muscle. Type 1 fiber, slow oxidative, fatigue resistant, and it has color red, high myoglobin content, low myosin ATPase. Type 2X is fast glycolytic and fast fatiguing, looks white, myoglobin low and very high myosin ATPase. Then phosphocreatine, this is the immediate ATP buffer, and it gives energy for 0 to 10 seconds, and the 100-meter sprint system is phosphocreatine. Then anaerobic glycolysis, glucose to lactate without oxygen. In this process you will find and 2 ATP per glucose from glycolysis, and it gives energy for 10 seconds to 2 minutes. Then oxidative phosphorylation, that means mitochondrial ATP production is happened or done by oxidative phosphorylation, and from this process you get about 30 ATP per glucose, and this is endurance system, that means it gets recruited last. Then calmodulin, this is the calcium sensor in smooth muscle, and smooth muscle doesn't have troponin, it has calmodulin, and it activates MLCK, that means myosin light chain kinase. Then MLCK, that we have said, is phosphorylates myosin to enable smooth muscle contraction. Then latch state, this is attached non-cycling cross bridges, tonic force at minimal ATP cost. Then intercalated disc, these are the connection between cardiac cell, and intercalated disc has gap junction plus desmosome, and it is only seen in cardiac muscle. Then funny current, this is the pacemaker depolarizing current, and this is responsible for SA node automaticity, and it has mixed sodium and potassium. Cardiac plateau, in cardiac action potential, this is the phase 2, and L-type calcium influx is responsible, and it prevents tetanus. And finally, the electrical foundation, we have resting potential, -85 to -90 millivolt in skeletal muscle, and it is set by potassium permeability maintained by the sodium-potassium pump. Sodium-potassium ATPase, 3 sodium out, 2 potassium in, 1 ATP used, and this maintains gradients, acts after the action potential and not during the action potential. And absolute refractory period, no stimulus can fire action potential in this time, and this is caused by sodium channel inactivation. And that is the whole lecture in 35 lines. If you can produce the hook for every one of those terms without looking, you're ready for anything the IMAT writes on muscles. Now let us prove it, because the next section is where all of this turns into marks. Everything we have built over the last 3 hours now get tested. 10 questions, IMAT style, and I'm going to solve every one of them out loud. Here is how I want you to use this section, and it matters. When each question appears, I will say pause and choose. You need to actually pause, actually commit to an answer. Do not watch me reason and nod along, because nodding along feels like learning, and it's not. The moment of commitment is where the learning happens, because that is when you find out what you actually believe rather than what you recognize. And I will not just give you the right answer. I will walk every wrong option and name why it is there, because every distractor in these questions is a trap we named earlier in the lecture. That is the whole point. Let us go. Question 1 reads, during skeletal muscle contraction, which band in the sarcomere does not change in length? The options are I band, H band, A band, distance between Z lines, distance between Z line and M line. Pause and choose. The keyword in the stem is the capitalized not. 4 of this change, 1 does not, and examiner has capitalized it because they know students skim. Go back to the interlaced fingers, that means the sarcomere. Remember, filaments slide and filaments never shorten. So anything defined by a filament length is fixed, and anything defined by a gap or a distance between structures will move. Option A, the I band. That is the region of thin filament with no overlap, seen here. As actin slides inward, like this, the region gets swallowed, it shortens. So eliminate option A. Option B, the H zone. That is the central gap where thin filaments have not yet reached, seen here. Sliding closes that gap, it shortens and can vanish entirely. So eliminate option B. Option C, the A band. This is defined by a full length of the thick filament. That means from here to here. Thick filaments do not change length. Therefore, the A band does not change length. Hold that one. Option D, the distance between Z lines. That is the definition of sarcomere length. If it did not change, there would be no contraction at all. So eliminate option D. Option E, the distance between a Z line and the M line. That is half a sarcomere, so it shortens by half as much as the whole. Still shortens. And notice how carefully the option is written to sound technical enough to be tempting. So after all this discussion, we eliminate option D and we are finally left with option C as the correct answer. And the lesson or the take-home message from this question is that A band is the immovable object in the sarcomere. If you try to memorize a list of which bands do what, you probably hesitated. If you reasoned from filaments flight, filaments never shorten. This took only 5 seconds for you. Question 2 reads, which molecule serves as the immediate calcium sensor in smooth muscle, triggering contraction? The options are troponin C, tropomyosin, calmodulin, ryanodine receptor, and Sarca. Pause and choose. The keywords are smooth muscle and calcium sensor. Both matter. Change smooth to skeletal and the answer changes completely, which is exactly why the word is there. Option A, troponin C. This is the correct answer for skeletal muscle, and it is deliberately placed first to catch the student who reads calcium sensor and stops reading. Smooth muscle has no troponin at all. So eliminate. Option B, tropomyosin, also part of the striated muscle system, and it does not bind calcium even there. It is moved by troponin, it is not the sensor. Eliminate. Option C, calmodulin. This is the free-floating calcium binding protein that activates MLCK in smooth muscle. Hold this option. Option D, ryanodine receptor. This is a calcium release channel on the sarcoplasmic reticulum. It releases calcium. It is not the sensor that responds to calcium to trigger contraction. Note also that smooth muscle has only a rudimentary sarcoplasmic reticulum. So, eliminate. Option E, Sarca. That is the pump that removes calcium during relaxation. It is the opposite end of the process. So, eliminate this too. So finally, the answer is option C, calmodulin. And what's the lesson? The trap here is the single word, smooth. Circle the tissue named in the stem before you look at the options. Because in this topic, the tissue determines the answer more often than the mechanism does. Moving to MCQ 3, it says skeletal muscle fiber is repeatedly stimulated at a frequency that does not allow relaxation between contraction. Which of the following correctly describes the resulting calcium concentration in the sarcoplasm? Pause and choose first. The stem is describing tetanus without using the word. Does not allow relaxation between contractions is the definition of fused tetanus. So first translate the stem into the concept. Option A, calcium concentration falls to resting levels between each stimulus. If that were true, the muscle would fully relax between stimuli, which directly contradicts the stem. Eliminate. Option B says calcium stays elevated because Sarca cannot keep up. That is precisely the mechanism we built. Pumping calcium back is an active process with a finite rate, and at high frequency the release outruns the pump. So hold that option. Option C says calcium rises and falls identically regardless of frequency. This is the classic wrong model where the student treats each stimulus as an isolated event. If that were true, summation could not exist. Eliminate. Option D says calcium is unaffected by frequency. Even stronger version of the same error that we made. So eliminate this one. Option E says calcium is released by the T-tubule membrane directly. This is a mechanism error rather than a logic error. T-tubules carry the action potential. The sarcoplasmic reticulum stores and releases the calcium. Eliminate. So option B is our correct answer. And what's the lesson? Notice that option B is the only one that mentions a rate-limited process. When a question asks why something accumulates, look for the option that names something that has a maximum speed. Accumulation always means production outran removal. Question 4 reads, rigor mortis, the stiffening of muscles after death, occurs because calcium floods the sarcoplasm from extracellular fluid. ATP depletion prevents myosin head detachment from actin. Troponin permanently binds calcium after death. Sarca pumps are hyperactivated, locking actin-myosin bonds. Option E, voltage-gated sodium channels remain permanently open. Pause and choose. When we say rigor mortis, this is a why question, and the answer is an absence rather than a presence. That is what makes it hard. 4 options describe something happening, and the correct one describes something stopping. Option A, calcium floods the sarcoplasm from extracellular fluid. That means calcium floods in from outside. Superficially plausible since dying cells do leak, but calcium alone would not lock the muscle. It would allow contraction while ATP lasted, then the muscle would still need ATP to detach. Calcium is not the mechanism of the stiffness. Eliminate. Option B, ATP depletion prevents detachment. This is our rigor mortis argument exactly. Hold it. Option C, troponin permanently binds calcium. Troponin binding is reversible and governed by concentration. There is no mechanism for permanent binding, and even if troponin stayed occupied, cross bridges would still cycle it if ATP were present. So eliminate this. Option D, Sarca hyperactivated. 2 errors in 1. Sarca needs ATP, so it cannot be hyperactive in a body with none, and Sarca removing calcium would promote relaxation, not locking. It states the opposite of its own consequence, so eliminate. Option E, sodium channels remain permanently open. This would concern membrane potential, not the mechanical state of cross bridges. It is answering a different question entirely. Eliminate. So finally, the answer would be option B. And the lesson, if you had ATP the wrong way around, believing ATP causes gripping, option A, C, and D would all looked possible, and B would have looked backwards. This question is a direct audit of whether your ATP model is inverted. Question 5 reads, which of the following correctly describes a structural feature that distinguishes cardiac muscle from skeletal muscle? And the options are, cardiac muscle fibers are multinucleate. Option B, cardiac muscle lacks striation. Option C, cardiac muscle cells are connected by intercalated disc containing gap junctions. Option D, cardiac muscle uses calmodulin as its calcium sensor. Option E, cardiac muscle lacks a sarcoplasmic reticulum. Pause and choose. So the STEM is asking for a feature that distinguishes cardiac from skeletal muscle. So the correct option must be both true of cardiac and false of skeletal. two conditions. Option A, cardiac fibers are multinucleate. False. That is the skeletal feature arising from myoblast fusion. Cardiac cells are uninucleate or occasionally binucleate. Option has swapped the tissues. Eliminate. Option B, cardiac muscle lacks striations. False. And this is the trap from slide 2. Cardiac muscle is striated. It is involuntary, which is a different property. Eliminate. Option C, intercalated disc containing gap junctions. True for cardiac and found nowhere else in the body. Both conditions satisfied it. Hold it. Option D, cardiac muscle uses calmodulin. False. Calmodulin is found in smooth muscle. Cardiac uses troponin C like skeletal. Eliminate. Option E, cardiac muscle lacks a sarcoplasmic reticulum. False. Cardiac muscle has sarcoplasmic reticulum and releases calcium from it via CICR, that is calcium-induced calcium release. The tissue with a rudimentary SR is smooth muscle, so eliminate this. So finally, option C is correct answer. What's the take-home message from this MCQ? The lesson is, notice that option D and E both describe smooth muscle wearing a cardiac label. Tissue swapping is the most common distractor technique in this entire topic. And the defense is simple. For every option, ask which tissue that statement is actually true of. Question 6 reads, in excitation-contraction coupling of skeletal muscle, what is the immediate consequence of dihydropyridine receptor activation? The options are given. Pause and choose. Two key phrases are here. First is skeletal muscle and immediate consequence. The word skeletal is doing the same job it did in question 2, and immediate rules out anything downstream. Option A, calcium influx from extracellular space. This is the cardiac mechanism planted in a skeletal question. In skeletal muscle, the DHPR is a voltage sensor that conducts nothing. This is the single most tempting distractor in the set. If you picked it, go back to the hand versus door image, so eliminate it. Option B, release of acetylcholine from acid. It doesn't make any sense on inspection. Acetylcholine is a neurotransmitter released from the nerve terminal, not from an intracellular calcium store. This is a free elimination. Eliminate. Option C, mechanical activation of ranitidine receptors on the SR. This is the direct mechanical coupling. DHPR changes shape and physically pulls RyR1 open. Remember, DHPR was connected with RyR1 like this. When DHPR was changing shape, RyR1 was getting opened. So hold it. Option D, activation of Sarca pump. Sarca operates during relaxation at the end of the cycle. Wrong direction and not immediate. Eliminate. Option E, phosphorylation of myosin light chains. Smooth muscle again. Skeletal muscle does not regulate contraction by phosphorylating myosin. Eliminate. So our final answer is option C. The lesson. This single question contains a cardiac distractor, a smooth muscle distractor, and a nonsense distractor. Once you can identify which tissue each wrong option belongs to, the question collapses in about 15 seconds. Question 7 reads, which event in the cross-bridge cycle is triggered by the release of inorganic phosphate? Pause and choose. This is the fine-grained question, and it separates students who learned the cycle as a rough story from those who learned it as an ordered mechanism. Option A, cross-bridge detachment. Detachment is triggered by new ATP binding, not by phosphate leaving. Different step entirely. Eliminate. Option B, ATP hydrolysis. This is what produces the phosphate in the first place. So it must come before phosphate release. You cannot release a product before you have made it. Eliminate on ordering alone. Option C, myosin head recocking. Recocking is powered by hydrolysis and happens while products stay bound. It precedes phosphate release. Eliminate. Option D, power stroke. Phosphate leaves and the head snaps through about 45 degrees. Hold it. You see here, when phosphate is released, the head is snapping back 45 degrees. So this is the answer, but we need to eliminate option E as well. So option E says calcium binding to troponin C. Calcium binding is upstream and independent of the nucleotide state of the myosin hat. It is what permits the cycle, not a step within it. So eliminate this. So answer is option D. Inorganic phosphate releases and it triggers the power stroke. ADP is released during the stroke after it has begun, and ATP binding causes detachment. The lesson? Two of this option, B and C, were eliminated purely by ordering logic without needing any additional knowledge. If an option describes something that must happen before the event in that stem, it cannot be caused by it. Sequence questions often contain free eliminations like that. Question 8 reads, a person is injected with a drug that inhibits the sodium-potassium ATPase in muscle cells. Which of the following is the most likely immediate consequence? 5 options are given. Pause and choose. Okay, the key phrase is most likely immediate consequence. And the word immediate is where students go wrong in the opposite direction from usual. They expect something dramatic and instant. Option A, muscles become incapable of generating any action potential. Too absolute and too fast. This is the water tower error. It assumes the pump generates the potential moment to moment. Gradients persist for a long time after the pump stops, so eliminate this. Option B. Intracellular sodium gradually rises and resting membrane potential becomes less negative. Note the word gradually. And note that less negative is correct technical description of depolarization. This matches the real physiology. Hold this. Option C. Calcium release from sarcoplasmic reticulum immediately blocked. The sodium-potassium pump, if I draw the cell, if this is the sodium-potassium pump, then the sodium-potassium pump has no direct role in sarcoplasmic reticulum calcium release. This calcium release is done by RyR. So unrelated system, eliminate. Option D. Acetylcholine degradation impaired. That would be acetylcholine esterase inhibition, a completely different drug target. This option belongs to a different question. Eliminate. Option E, myosin ATPase activity is directly inhibited. Two different ATPases sharing part of a name. The sodium-potassium ATPase is a membrane pump you see here. Myosin ATPase is in the motor hat. If this is myosin hat, then ATPase will be in the motor hat. Inhibiting one does not inhibit the other, so eliminate that one. That leaves us with option B as our final answer, that the pump maintains gradients rather than generating the potential directly, so inhibition causes a gradual rise in intracellular sodium and progressively less negative resting potential. What are the lessons? So option A and option B describe the same general direction, but A overstates the speed and severity. When two options point the same way and differ in magnitude, the more measured one is usually correct, because physiology has buffers and reserves almost everywhere. Question 9 reads, which fiber type would predominate in the soleus muscle of a marathon runner's leg and why? The options are given below. Pause and choose. You see the soleus muscle here? The soleus muscle is in the posterior segment of our leg. And if you see the options, every option here has two clauses joined by because here, and both clauses must be true. This is the structure I flagged earlier, and this question is the reason I flagged it. Option A, type 2X fibers, because they produce the most ATP per glucose. Both clauses are wrong here. Wrong fiber type for endurance, and type 2X uses anaerobic glycolysis, which yields 2 ATP per glucose, the lowest of any system, not the highest. So eliminate. Option B, type 1, because they are fatigue resistant and primarily oxidative. Fiber type is correct for an endurance postural muscle. Reason correct. High mitochondria, high myoglobin, oxidative metabolism, high fatigue resistance, and both clauses true. Hold it. Option C, type 2A, because they are the only type capable of using fatty acids. The word only should make you suspicious immediately. Type 1 fibers use fatty acids extensively. False exclusivity claim. Eliminate. Option D, type 2X fibers, because anaerobic glycolysis is the fastest ATP source. The second clause is arguably true in terms of rate, but the first clause is wrong for a marathon runner. A true reason attached to a false conclusion is still a wrong answer. Eliminate. Option E. Type 1 because they have the highest myosin ATPase activity. And here's the trap. The fiber type is correct. The reason is exactly backwards. Type 1 has the lowest myosin ATPase activity, which is why it contracts slowly. So eliminate this one. So our final answer is option B. And the lesson, option E is best designed distractor in this entire set because it gives you the right answer with the wrong reason. Students scanning for type 1 ticket and lose the mark. Always read past the fiber type to the justification. Question 10 reads, which of the following statements about the action potential and cardiac muscle is correct? Options are given here. Pause for a while and choose. Then look for my explanation. Okay, so 4 false statements and 1 true one. So this is a pure recall check across everything in slides 12 and 14. So option A says it lasts 1 to 2 milliseconds, identical to skeletal muscle. False. That is the skeletal duration. Cardiac lasts 200 to 400 milliseconds. Eliminate. Option B. Plateau phase caused by prolonged potassium influx. 2 errors stacked here. The plateau is calcium, not potassium, and potassium moves outward, not inward, because it is concentrated inside. Even the direction is wrong. Eliminate. Option C. The long absolute refractory period prevents tetanic contraction. True, and it is the central functional fact about cardiac muscle. Hold it. Option D. Pacemaker cells depolarize due to sustained sodium influx. Wrong direction again. Efflux of positive charge would hyperpolarize, not depolarize. The funny current is an inward current. Eliminate on direction alone without even needing the name. Option E. Gap junctions are not involved in propagation. This is the exact opposite of the truth. Gap junctions are the entire reason the myocardium behaves as a syncytium. Eliminate. So our answer is option C. And the lesson? Option B and option D were both eliminable purely by checking the direction of ion movement against the concentration gradients from slide 13. If an option has an ion moving the wrong way, you can discard it without knowing anything else about the topic. Every lecture in this series is on the channel, free along with past paper analysis and the full application walkthrough. Subscribe so the next one finds you. If you want the written guides, they are at myelinhq.gumroad.com. And if you spot an error anywhere in my material, write to me at tutor@myelinhq.com. I fix it and everyone gets the update for free. Look back at what just happened across 10 questions because there is a pattern and the pattern is the real lesson. Not one of those questions required a fact that I had not already taught you. Every single wrong option was a trap named earlier in this lecture. The A band does not change. Troponin missing from smooth muscle. Cardiac DHPR planted in a skeletal question. ATP for detachment rather than attachment. Phosphate rather than ATP driving the power stroke. The pump that maintains rather than generates. Type 1 fibers with low rather than high ATPase. This is what the IMAT is actually testing, not whether you have read more, whether you can eliminate a well-designed distractor. The examiner is not trying to find out if you have heard of the sarcomere. They're trying to find out whether you understand it well enough to reject something that sounds right. And notice how often the elimination came from a structural check rather than deep recall. Which tissue is this option actually describing? Which direction is that ion moving? Does this step have to happen before the one in the stem? Are both halves of this because statement true? 4 questions, and between them they killed most of the distractors in this set. So here's what I want you to do before the next lecture. Go back to slide 15, cover the right-hand column and work through all 35 books out loud. Anywhere you hesitate, that is not a gap in memory, that is a trap with your name on it, waiting on exam day. Everything in this lecture, the master note, the anchor slides, and the full question bank is available through the link below. Next lecture, we move to the nervous system, where the resting potential and action potential we built today become the foundation for everything. Thank you for watching. If you like the video, please like, drop a comment, subscribe. I will see you there in the next lecture. Peace.